Testing, testing. Okay, good morning, everybody. It is about 9, so we'll get started. So just to remind you of the format for today, so we're going to have 3 sessions where we've got a couple of speakers starting off, and then we're going to lead those into panel discussions. So we'll invite our panelists to come up and sit at the table here with our moderators and presenters. And to just have an informal discussion both on the points that were presented in each of the presentations, but then also anything else that the panelists want to bring up. We will, so we met a lot of folks yesterday in the lightning talks, but I think for the panel members, when we start the panel session, if we can just start with a short introduction from each one of you so we know who's up here and what type of collections and research they represent, that would be helpful. And then after about a half an hour of panel discussion, or if the group has burning questions, we'll progress into a full group discussion. So very similar to what we saw yesterday with the discussions in between the different lightning talks and that sort of thing. All right, so any questions to start off with? Those are the general rules of the road. So for this first session on varieties of food, Martin's going to be our chair for this. He'll introduce the speakers, and then the panelists that I have include George, Savi, and Patricia. So I'm going to see Savi. Is George here? Okay, so George isn't here. Maybe he'll show up. All right, thanks.
Again, I'm Martin Kalfatovic with Smithsonian Libraries and the Biodiversity Heritage Library, and I'm going to introduce our 2 panel speakers today. Our first speaker is Rod Page. Rod's a professor of taxonomy at the University of Glasgow and is also the current GBIF Science Committee chair. He's a lapsed taxonomist who, after working in phylogenetics, moved on to biodiversity informatics. When not hacking code, he writes blog posts on why everything in the field sucks. Rod?
Thanks very much, Martin. So yes, if you want to read why everything sucks, there's a blog post up there and a Twitter handle there. I have a really difficult time coming up with titles for talks. So I've come up with 3 titles: Unknown Knowns, Long Tails, and Long Data. So what I want to start with is I want to unpack what those 3 phrases mean. So many gadgets, so many buttons. So the first one, the unknown knowns, is kind of a homage to Donald Rumsfeld. So in the first Gulf War, he had this amazing kind of press conference style, and he gave this much quoted phrase about there being known knowns, known unknowns, and unknown unknowns. And I think what he was trying to do is say basically there are three kinds of things. There are things you know, there are things you know you don't know, and then there are things you don't know that you don't know. And they're the things that cause you the most grief. But he missed one really key category. So we know this, we know we don't know that. We have no idea what's going on down here. The stuff here is what I'm interested in. This is the stuff that we know without knowing it. Now, what I mean by that is if you imagine all the data locked up in the scientific literature and the grey literature and databases and collections and all kinds of different places that are not connected. So if we could add all that together, we'd probably know a lot more than we think we know. If you think about, we've had about 2 centuries of scientific research in this field, we probably know vastly more than we actually know. Whoa, too many buttons again. The second title I come up with is this idea of long data. This is a blatant steal from a wonderful book called Uncharted. It's a book about the Google Books project. And one of the outcomes of this book was this notion of long data and looking at data over time. So the sort of most practical example of this is this wonderful tool, the Google Ngram Viewer. Hands up those who've come across the Ngram Viewer. It's great fun. So basically what they've done is they've scanned all the books they could get their hands on, and then they looked at the frequency of words over time. And you can do some fun stuff. So what you're seeing here, this is 1800, this is 2000, this is biodiversity, clearly the place to be, taken off. And here's food security, which is not really doing much at all. So based on that, biodiversity rocks and food security, not so much. To put this into a bit of context, I then threw in agriculture. This is a bit of an eye-opener. So here's biodiversity now dwarfed by mentions of agriculture in these scanned books. So you can get some really interesting insight into changes in word frequency over time, what people are interested in. There are some big caveats with this approach, the biggest one being that you're sampling over time and you're hoping that these samples are kind of representative. In the world of open access, in the world of open access to biodiversity literature, this is not the case. This is kind of my attempt to summarize the Biodiversity Heritage Library, a sort of small subset of it. One of the projects I work on is something called BioStore. BioStore's mission is to find articles in BHL, so convert it from scanned books to scientific articles that you can cite. And the distribution of articles over time is distinctly bimodal. This is Mickey Mouse. Mickey Mouse is evil. Because of Mickey Mouse and the Walt Disney Corporation, the US copyright, we had a really hard rule here in 1923. Prior to that, this is pretty much all out of copyright. You can do with it what you want. After that point, you're in trouble. And this is a major sort of chilling effect on our access to biodiversity literature. All the stuff over here is basically content which BHL has had to negotiate individually with publishers, museums and collections. Can we scan your content? Prior to that, you can just do whatever you want. So clearly, if you're going to look at trends in anything you're interested in over time in this literature, you've got to be aware that you're going to be missing a huge chunk of information from the 20th century. The final title I sort of was struggling with is this idea of the long tail, which you've kind of heard already before. This is the 80-20 rule. What you're seeing here is a very, very common pattern of a situation where some things have lots and most things have very little. You can apply this to just about anything. For example, money. There are a few incredibly rich people and the vast majority of us are basically poor or not really rich. What you're seeing on this diagram is this is a quick analysis of Wikipedia. So for every mammal species in Wikipedia, I've measured how big is the page, just counted the characters. And what you find is that you have a few really large articles. Pandas, chimpanzees, humans, lions, people care about those. But there's something like 6,000 or 7,000 extant species of mammal. The vast majority of those have very, very short pages. It's a mammal. What more do you want from me? And this is the kind of sort of situation you see. So we have this thing called the long tail, this sort of very long thing down here. Now, I think this is one way of making a case for the taxonomic literature. I would argue that for the vast majority of species on the planet, the only place you can go to read anything about what they are, where they are, what they look like, what they do, what they eat, what eats them, is going to be the taxonomic literature, perhaps just the original description, because that's the only time anybody has looked at it and written anything about it. So taxonomy kind of lives down here. Okay, so having set the scene, I want to give you a couple of examples of what I think we can do. So the idea here is that not only are we interested in literature as something for humans to read, but we want to let machines loose on it. We want to sort of text mine it. So let me show you a couple of examples of the kind of things you can do when you let the machines at this kind of literature. So there are a couple of things you can do if you're just interested in, say, looking for taxonomic names in the literature. And here's a couple of examples. So this one on the left, probably slightly hard to see. This is a set of taxonomic names from one particular paper. And you'll see they're kind of clustered into two groups. These things up here, these are shrews and rats and mice. These things down here are insects, and actually they turn out to be fleas. So this paper is clearly about host-parasite relationships between mammals and fleas. You can sort of extend this to not just individual papers, but to whole literature and to whole databases. So the diagram on the right, this is not the scientific literature, this is GenBank, giant database of DNA sequences. Turns out if somebody sequences an insect, very often they will tell you what plant or what other organism the insect was found on. So GenBank, what we think of as mainly a database of sequences, is actually an ecological database. And what you're seeing here, these are bacteria, green plants, fungi, animals. Vast majority of these connections between insects involve green plants. These could be pollinators. These could be parasitoid relationships with other insects on those plants, or direct kind of herbivory relationships. And there are a few things here. These are connecting insects with vertebrates. These are typically host-parasite relationships. Just one more example of this. Oh, no, sorry, two more examples. You can also query the literature not just from scientific names, but from geography. This is another sort of little display from BioStore. What you're seeing here is for every article that I've taken out of BHL and put into BioStore, I look for latitude and longitude pairs. And if the paper mentions a particular place, I'm not just using place names, it's actually latitude and longitude, you get a dot on the map. And this is an interactive map that you can zoom in, you can draw a box around, you can get a list of papers from a particular area. So now if your focus is a geographic region, not necessarily a species, you could say zoom in to, I don't know, some sort of protected rainforest in Peru and ask the question, what do we know about that? And if somebody's mentioned that locality in a paper, you get a list of papers and you can go and discover some information. So again, the first bit of this is all done by machine. We find locations in the text. The final example, this is not BHL, it's not anything I've been involved in. This is a really cool piece of work by Paul Oldham and colleagues. What Paul did was he data mined the patents literature. So basically he went through all the patents and searched for taxonomic names of species. And then he went to GBIF to find out where in the world these species were. And lots of kind of diagrams, but the key point here is that he discovered that essentially 4% of the species that are described, which of course is an even smaller fraction of what's actually out there, appeared on these patents. So his argument was that if you think of biodiversity as something you want to exploit rather than conserve necessarily, we're doing a really very poor job. Only 4% of it has appeared on these patents. Also geographically, it's incredibly biased. If we ask where these species are, there's a whole sort of area in the tropics, not a great deal is happening. The vast majority of species that are patents, covered by patents, are North America and in Europe. So this is a slightly interesting from perspective of, say, GBIF, where I guess one of the rationales for GBIF is let's conserve the planet, let's find out where everything is. This is flipping that completely around. Let's exploit the planet as much as we can. But it's a really interesting example of what you can do with some decent kind of text mining. Okay. So the final thing I want to very briefly touch on is something called the biodiversity knowledge graph, which is a term that's a blatant ripoff from Google, who've been sort of promoting this idea of a knowledge graph, I guess, the last 5 to 10 years. This is kind of a cartoon of the sort of standard things that most people working in biodiversity and I guess taxonomy and systematics, that sort of side of biodiversity, care about. We care about species, taxonomic names, the literature, taxonomists and other researchers. We care about specimens. We care about where they come from. Have you got any DNA from them? Can you build a tree? What kind of characteristics do they have? And so on. And the ultimate goal would be, wouldn't it be wonderful if we could link all this stuff together? So that if I'm writing a paper, I could directly link to not only other papers, which is what we do now, but to the specimens, and from the specimens to where they're found, and to the sequences into the trees, and join it all up. And at the moment, this is incredibly hard to do. I want to sort of finish up by giving you one example, of a fairly trivial kind of example, the kind of things you can do if you can kind of connect things like publications, specimens, and localities. So this is not a particularly food security or agricultural example, it's more a kind of disease example, but bear with me. This is a paper in PubMed, which I guess most of us have come across, these sort of major biomedical literature database. And here's a paper, and it's about some snails, which carry a fairly nasty kind of disease that affects people. And if you're in PubMed, PubMed have a nice controlled vocabulary that tells you what the paper's about. It's about disease vectors, it's about ecology, it's got different genders in it, and it's got the name of the disease, and so on. What PubMed doesn't do in this case, it doesn't tell you where you can read the paper. So BHL and BioStore fix that for you. Here's that paper, it's openly available, it's open access, it's been scanned by BHL, it's in BioStore. So as a person, you can go and read it, but machines can also read it. And what machines have done is find out, for example, that this little dot here, that's where in the world we're talking about, because the paper actually mentions that locality. Let's maybe zoom in a bit. So there are a couple of dots here. These are the localities that the paper is talking about. And also embedded in that text are some cryptic little codes, these ANSP things. These are specimen codes. These tell you that somewhere in a collection, in this case I think it's in Philadelphia, you can find those snails. And these snails, because they're in a collection that's been digitized, are also in GBIF. And so you can go and have a look at those. And this is a fairly old example of what GBIF looks like. Looks a bit more swish now. So now we can actually go and have a look at those specimens in GBIF and put them on a map and learn about them. So the point of this is we've gone from a literature database, mainly focused on the biomedical literature, to open access version of that paper from BHL in the middle, to a specimen database, GBIF. Now, once you can do that, there's a number of things you can do. So at the moment, these things are all fairly kind of separate. GBIF is the database of where things are in the world. It's dots on maps from museums, herbaria, and citizen science projects. PubMed is the database about biomedical literature. You could connect these. So for example, you could imagine that given we've made those links, you can now query PubMed geospatially. You actually know where in the world that paper is very precisely. So you could start to query that database using the spatial coordinates you've got from the specimens that are in GBIF. Likewise, you could look at GBIF as not just a database about museum records and citizen science, but you could query it as a kind of biomedical database, because any term that's linked to that paper here applies to these records in GBIF. You want to find about this particular disease, you can actually go to GBIF and say, okay, here's a PubMed term for that disease. I should go query here because you've made those links. So the argument is that once we connect these things together, we can do things that are way more sophisticated than what we're allowed to do or enabled to do at the moment. Okay, so by way of summary. So first point is the open access literature, which is in the biodiversity world is mostly but not exclusively BHL. There's huge potential there for text mining this information. It's long data, it spans a long period of time, and because a lot of it is fairly obscure in a sense, it's the long tail. And this is going to be fundamentally important for finding out about things that are not the kind of tiny fraction of model organisms or things we currently use for food. Huge scope for text mining. There's all kinds of stuff that you can find. Some of the stuff we can do right now, things like scientific names, it's pretty straightforward. There's really good methods for doing that. You can find places, you can find specimens. It's a bit harder to find attributes of organisms. There's been some quite interesting work done, particularly in botany, about how you'd read as a computer a description of a plant to figure out how big it is, where it grows, what it sort of does. And ironically, despite all our sophistication, search, in other words, finding stuff, is still the killer app. It's still one of the greatest challenges we find is just finding stuff. And the final thing, and sort of holy grail for people like me who are interested in this, is linking things together. And I think this is one way we're going to tackle these unknown knowns. We have the situation of all this kind of disparate information and different papers and different data sets and different collections. And if we join the dots and connect them up, then we're going to discover, I think, many of those unknown knowns. Thanks so much for your time.
Thanks, Rod. And we'll have questions at the end when we get the rest of the panel up. Our next speaker is Dr. Ari Novi. Ari is the director of the U.S. Botanic Garden since 2012, not 1912, and was appointed director in 2014. In this role, he leads the garden's efforts to demonstrate the aesthetic, cultural, and economic, therapeutic, and ecological importance of plants to the well-being of humankind. He received his Bachelor of Arts from NYU in Italian and his doctorate from Rutgers in plant biology. He remains an active researcher, holding an appointment as a research collaborator at the Smithsonian, National Museum of Natural History, Botany Department, and has a deep passion for translating science into best management practices and public education. Ari?
Morning, everybody. Thank you so much for having me. I'm going to give a little bit of an overview of botanic gardens in general as institutions. So I'm speaking here not only as the director of the local botanic garden, here, the U.S. Botanic Garden right at the foot of Capitol Hill. I'm also on the International Advisory Panel for Botanic Garden Conservation International, which is located at Kew, but also has some satellite organizations, including in the U.S. And that organization is very much the clearinghouse for botanical information worldwide, and has a very strong focus currently on crop wild relatives, quite closely related to the Millennium Seed Bank, which we heard a lot about yesterday. So I want to talk a little bit about botanic gardens in general, what we do. You heard some great examples yesterday, so this will be a little bit of recapitulation. Give a couple of examples, talk a little bit about what's the idea, why are we involved in food security, how should we be? And then I would be really remiss if I didn't spend a pretty fair amount of time talking about educational outreach. A lot of what we do here as scientists, data mining, building collections, thinking about how they can be used to solve a variety of the world's problems, really will not amount to much if the public isn't heavily behind what we're doing. I think in some of the chatter I've been hearing yesterday and today, I don't think there's a single person here who doesn't lament the lack of funding for their individual area of research and for our endeavors as a whole, and public outreach and how we do that and how our collections can accomplish that is a critical aspect of what we do. So to start with, what are botanic gardens for the non-plant people in the room? Here's ours. They are essentially organized collections of plants. I mean, that's really the only way to very, very simply define them. In fact, you can be a botanic garden, self-certified as a botanic garden, so long as you have a collection of plants that is properly labeled, and properly isn't even defined. But historically, they're very connected to both plant utility. The first botanical gardens, at least in Europe, were related to especially medicinal plants and other useful plants, dyes, poisons, things like that. And then the idea of phylogeny, the study of comparative plant, first anatomy and morphology, and of course later on genetics, to figure out the relationships between plants is quite important. But botanic gardens are a lot more than that. Of course, they are the major botanic gardens of the world. Are also the locations of the major herbaria of the world. That's not always the case, but that is very, very predominantly the case. And the herbarium record are the natural history records that really do indicate where plants are found throughout the world, as well, of course, as the isotopes for plant naming, type specimens for plant naming, and really the authoritative source for both the genetics and the taxonomy of plants. This is the National Herbarium here in Washington, D.C. at the Smithsonian. Botanic gardens also have collections. Seed banks are one of them. We heard a lot about Kew yesterday, so I won't go into that, but many, many botanical gardens do maintain seed banks. And then botanic gardens contain living collections, and whether you consider seeds a living collection or not is sort of just up to your organization, but many consider them living collections. What I'm showing here is a picture both of part of the orchid collection at the U.S. Botanic Garden, but underneath it, this is part of the National Kiwi Collection, which is at Wuhan Botanical Garden in China. And so there are many, many botanical gardens which contain very important worldwide collections of agronomically important species. This is a very expansive network. Just within the BGCI database, there are over 3,300 botanical gardens worldwide. You'll notice they are very nicely distributed in accordance with population. So the botanic gardens tend to be where the people are. And of course, many, many, many specimens come from other parts of the world, but the gardens are where the people are. Just within the U.S., we have over 750 institutions that are considered botanic gardens. And once again, it's basically the same as if you were to do a population density map. Why? Why do we do this? It's really important to recognize that botanic gardens getting involved in agriculture is absolutely nothing new. In fact, I think a very strong argument can be made that before, in the U.S., at least the 1860s, when the U.S. Department of Agriculture was created, and certainly later in the 1800s, when other countries begin to develop their ministries of agriculture, botanical gardens are actually some of the prime locations for international agricultural extension. The colonial system of European botanical gardens was set up very much for the technology transfer of agronomically important plants. Plants like eventually tea, many spices, rubber, cinchona, which brings us quinine, sisal, all kinds of plants that are very useful in agronomy were being moved from one tropical part to another tropical part of colonial empires. And it's really a very strong reason why the various monarchies supported botanical gardens early on in European history. So we're not talking about anything new. If anything, we're really talking about a rediscovery of an important connection with botanical gardens and botanical institutions and an agronomic past that has been separated for about 100 and some odd years at this point. But it's important to recognize botanical gardens, as I mentioned earlier, are the repositories both of the information and raw samples for plant biodiversity worldwide. We all probably know, and there's a couple of slides in here I stole from Paul Smith, who's now at Botanic Garden Conservation International, but used to be with the Millennium Seed Bank. This is one of them. So globally, about 80% of our plant-based calories come from just 12 domesticated plant species, 8 cereals, and about 4 tubers. About 50% of our total calories come from wheat, rice, maize, and if you add in really soy to get meat, that's pretty much 80% or so. So we're using a very, very small amount of the available biodiversity for food, but there's at least 12,000 edible plant species. And there's about 20% of the plant species that are currently threatened with extinction. That's of all plant species, and that 20% number is from the latest Kew state of the plants in the world. And this is really important for 2 reasons. One is the direct reason. Okay, if we're interested in beginning to exploit these 12,000 really underutilized species, which have tremendous potential, especially with such a high dependence on a small number of crops, we better save and save wide germplasm of those plants, especially the ones that are in danger of going extinct or becoming at least very seriously genetically paupering. But secondly, and maybe even more importantly, the threats to plant biodiversity worldwide are primarily agricultural. And now this is also from the State of Plants report from last year. If this was done in Europe, but if this was done in the U.S., we consider logging part of agriculture. So actually, about 50% of challenges to plant biodiversity worldwide are due to agriculture. So we have this really interesting dual relationship where botanical gardens, which are the sort of protectors and studiers of plant biodiversity, are both very interested in being relevant to solving societal problems such as food insecurity, but at the same time, on the other side of our mission, which is to safeguard plant biodiversity writ large, agriculture is the greatest threat to that plant biodiversity. And so now I don't see any contradiction whatsoever in those things, but it's important to daylight this, and it's important to really realize what we're doing. In fact, the better we are at more sustainably and effectively farming, hopefully the less we can do some of these activities on here that are so threatening to plants. So with that, sort of setting the table, I just want to give a couple of examples in addition to the ones that we've heard about yesterday. I'd like to mention the National Tropical Botanical Gardens, which are primarily in Hawaii, although there's one in Florida in the United States. They maintain the World Breadfruit Collection, and here's a picture of that here. This is I'm not going to talk really at all about the Crop Trust, but this is a Crop Trust site as well, and the Crop Trust is a very important kind of connecting institution that was mentioned yesterday as well. But this is an example of a botanical garden directly involved in plant collection and breeding, just like we saw in Wuhan with the kiwi collection. Another great example and really big focus right now within the community is collection of germplasm of crop wild relatives. This is a graphic I took from Bioversity, which is a part of the UN, part of CGIAR system. And for those who don't know what crop wild relatives are, those are the relatives of crops that can be, in one way or another, have genes integressed into crop species that are of importance. And they're very, very important because they may contain And in many cases, we've already found this. For example, a great example is the current downy mildew disease on basil. The resistance is coming from another basil species that was found in a basil germplasm repository. But pest and disease resistance, nutritional quality, and all kinds of environmental tolerances, those genes may be in primary, secondary, tertiary gene pools related to the crops. It's estimated that $115 to $120 billion of value resides in crop wild relatives. And these are under threat. We have to go after them. We saw this graphic yesterday, and I really want to mention it again here because this graphic was populated by deep research into herbarium species. So without the botanical gardens maintaining their herbaria and other geographic locale information related to plants, we couldn't even go about beginning to set priorities, hotspot targets, and things like that for collecting for agronomic purposes of these crop wild relatives. Another great example here, this is a collaboration between the Land Institute, which is in Salina, Kansas, and Missouri Botanic Garden in Missouri. And what they're doing at the Land Institute is they're really trying to breed perennial grain crops. The idea being that the root structures are much, much deeper so they can access more nutrients. You see here some perennial species next to annual wheat throughout the year, and you can sort of see for yourself what's going on. But they recognize they don't have all the traits that they need in the plants that they're targeting, and so they've forged a collaboration with Missouri Botanic Garden to identify the compatible crop wild relatives with the species that they're targeting for seed production and to get all the agronomic traits in there that they can possibly help out. So this is a very important and interesting. So here's a great example of an agronomic institution partnering with a botanical garden, pulling off of all that great collection-based information. Another quick one from Missouri Botanic Garden. I love reminding all of my European friends in the room that if it wasn't for the American grapevine, there'd be no more European grapevine. Back in the late 1800s, an insect called Phylloxera decimated European grapevines, and it was the answer was American rootstock. So Missouri Botanic Garden is continuing to investigate and pull land races of American grape rootstocks, primarily species like Vitis labrusca, which have resistance to this still insect problem as an important rootstock for the grape industry. And then in terms of, I'm running out of time here, but in terms of outreach, the really big issue is, and this is a general problem in biodiversity for everybody, right? We are where we are in cities. We're in an urbanizing population. The United States is 80% urbanized at this point. The world is about 60%, and both of those numbers continue to rise. So we're where we are. Here's a population density map of the United States, but maybe better is this map of North America that you see all these wonderful ecosystem variety in North America. And if you can see, there's some red dots kind of where the cities are. Those are urban areas. In the United States, at least about 4% of the United States is urban. About 50% of the land area of the United States, including Alaska, is agricultural lands, including rangeland. So the reality is people are in the cities and biodiversity and agriculture are somewhere else, which means we have a tremendous responsibility, and our collections are a tremendous tool that can be leveraged to bring that information to people. And so I want to just run through a bunch of examples. I mentioned this earlier. In the US, we're over 50% agriculture. The new census should be out this year. I'm very excited for the new data, but this is the data I have right now. Here's the US Botanic Garden. We've been focusing for many years now, about 3 in a row, really trying to hit hard on teaching urban populations about the importance of food. And you start simple, and many botanic gardens do this. This is an exhibit we have just basically on food, recognizing that people have no idea what the plants look like that their food actually comes from. Here's okra. We're talking here about plants that are important from Africa, that were brought with diaspora populations, in this case slave populations, and have become important food sources in the United States. Here's cabbage. People don't really know what cabbage is. They have no idea what the varieties of cabbages are, that it's started as a little tiny mustard cress and all that kind of a thing. Chicory, beets, tomatoes, and I mentioned cabbage before, and I've always loved this diagram. You know, if you think morphologically, you really don't connect crops because they've been so highly derived through artificial selection from their wild relatives and their crop wild relatives. And brassicas are probably one of the best examples of that. All the various brassica forms we have, which are all the same species, kohlrabi, kale, Brussels sprouts, and all the cabbages and broccoli are all Brassica oleracea. In the wild, it looks like this little mustard weed. So we actually recapitulated this with plants, which was really powerful for people. And then we related them to dog breeds because people understand dog breeds. It's as Same story. We also embrace controversy. We grow genetically modified plants. Not a lot of botanic gardens do. I don't know why they don't. Perfectly legal. Here's BT cotton at the U.S. Botanic Garden. Great area of entry. Also, well, we've been doing a couple more exhibits. One we did 2 years ago was called Amber Waves of Brain. The patriotic titles kind of write themselves when your botanic garden is right at the base of Capitol Hill. Here we were celebrating the 100th, what would have been the 100th year of Norman Borlaug's birth. And Norman Borlaug, of course, won the Nobel Peace Prize, for saving 1 billion people from starvation, according to the Nobel Peace Prize Foundation, mainly through his wheat breeding efforts at CIMMYT, which is the CG Center, it eventually became the CG Center for the International Research of Maize and Wheat. And what we wanted to do is we wanted to showcase the varieties and the progenitor varieties of wheat that he developed that helped stave off so much starvation, as well as talk in general about wheat as an agricultural commodity crop. So we planted a series of about 25 varieties that showed about 20 years of progress in his breeding program, and we were careful to include the wheat. I don't know any wheat breeders in the room. Nora 9, right? The very important Japanese variety that came through the University of Washington in the late 1800s, which contained the semi-dwarfing variety that was really key to that breeding program that's still around. We got all these plants from the USDA Germplasm Repository in Idaho. They were grown here in Beltsville because some of them needed vernalization, which we didn't have the ability to do at the U.S. Botanic Garden, and then we planted them out. At the same time, in March, we put up an exhibit here that talked about this kind of stuff before the growing season got going, including, and you'll see somebody talk yesterday about UG99, horrible rust strain, what's going on today, and starting to explain why do we breed plants? Why do we look for these varieties, for these genes that confer all kinds of important agronomic traits? And this is an important thing to do. Here's a schematic of the bed. Here's what it looked like. These are all the 30 varieties of Borlaug's program going in. As they grew up, you could see tremendous morphological variety, even in wheat. So even the uninitiated folks who aren't used to looking at agronomic crops, well, you can tell the difference between a dwarf, semi-dwarf, and tall wheat variety on list. There's actually a lot to look at, and once you start to focus your eyes in on that, people can really see that. Here's a couple different examples. Lodging was a horrible problem with wheat. You know, it would fall down in the fields and then it would rot. Borlaug was breeding to get dwarfism. So here you see varieties early on in the program that weren't yet dwarfed and varieties later on. And the wind cooperated and knocked over the tall ones, and boom, we can tell our story. Here we also grew the 6 major classes of wheat. I won't go into the 6 major classes, but those are the commercial varieties of wheat, the classes of wheat that are useful because they make different kinds of wheat products. This is one of my favorite shots. These are the ancient progenitors of wheat, so einkorn, emmer, and spelt, as well as durum wheat, which is pasta wheat. I like this shot because this is a common garden experiment. It's a little hard to see here, but you'll notice that the wheat all the way on your left is a little bit less green and a little bit shorter, and that's durum. And that's because genetically, durum wheat has a decreased ability to take up phosphorus, which is part of why it's a little bit more expensive and part of why really good pasta using 100% durum wheat is a little bit more expensive than other kinds of pasta. And this is a common garden. This soil were treated the exact same, and these are opportunities to explain to people all of these agronomic principles and the genetic diversity that impacts agronomy based on all of these characteristics of the environment. Basically, we're teaching people G by E without really mentioning technical terms. And we did what farmers do afterwards. When the wheat was done, we went to soybean. Half organic, half GM, can't tell the difference. We also did this great thing. We built a wheat sculpture, and we populated this bed with weeds of wheat. These are all important agronomic weeds of wheat. Weeds can reduce crops by 50% in some cases, including wheat. In many cases, wheat is its own weediest species, but really getting at the public what's important, what's really going on. This doesn't relate so much to germplasm, although important collections of weeds are very, very important for a lot of study areas. And then the last exhibit we did related to this is called Exposed, The Secret Life of Roots. And here what we did was we were able to work with some scientists who'd found a way to grow tall grass prairie plants and agronomic plants in a way that you could extract the entire root systems without damaging them. We fixed them in glycerin, and then we displayed them against the picture of the root zone of the ecosystem where these came from. Now, we did this for a very specific reason. We chose to do tall grass prairie plants because the tall grass prairie is essentially what has built the soil that the United States Midwest agriculture is based on, that part of the breadbasket of our country. So it's hard to see here, but on the left side, these are annual plants like wheat, and then in the center, you get some of the native species that over millennia built up this really rich soil profile that is also partially that is very responsible for the success of a lot of American agriculture. Hard to see, but on the far side, there's actually soy down there too, and with the roots exposed, you can see the nodulation and all the kind of interesting things happening underground in the soil. And this is a really rich experience. People are rarely brought into contact with these parts of plants in a way that's so dramatic. And it was an incredibly powerful and wonderful experience for everybody. And I'll add that both of these, the three exhibits I've shown you, each had about 600,000 visitors. The wheat exhibit as well was redone by the Kansas State Fair and was seen by another 10 or 20,000 visitors there as well. So you can make a pretty deep impact and you can really educate a lot of people in a relatively short period of time with some really wise usages of your germplasm repositories in an outreach scenario. So just very quickly, some of the challenges that I think we really are going to continue to face as we look at these problems. There's really not enough cross collaboration, especially with agronomists and breeders. Agronomists and breeders jumped to help us with this stuff because no one's asking them. They're like, really? We can do this. We can get this information out to hundreds of thousands of people. But the CropWild relative research, Kew's doing a great job, but most other gardens are not yet engaged deeply with their agronomic folks to understand what the targets should be. And that's a great area of research that we need to go on. Another big problem is a lot of, even gardens, even botanic gardens that operate internationally, have a very incomplete understanding of international laws and regimes. We need a lot more workshops on the CBD, the Nagoya Protocol, the International Treaty on Plant Germplasm, and UPOB, and all the other things that come out to weigh in. We need lawyers, unfortunately. Herbarium specimens are still not fully digitized. There's no good source to search the world's herbaria. You can get, a lot of the type specimens are, a lot of the big herbaria have some availability to cross look, but really it's hard slogging out there if you want to go and really search the world's herbaria. We still haven't even named everything, so that's a big part of it as well. Other plants were better than some other categories. Nematodes, yeah, we just learned yesterday, only about 10 or 20% of the name. And also botanic gardens are a pretty diffuse network, mostly of pretty small institutions. In the US, you're a big botanic garden if you have more than a $3 million budget, which means you don't have a huge budget to bring lawyers in and teach you about international treaties and things like that. So this is a very, very serious problem. There's a small number of large gardens like Kew, like Missouri Botanic Garden, New York Botanic Garden, that are kind of leading the charge on this, but it's really not a critical mass enough worldwide. Now then, my last slide, just to let you know about a couple of resources for those who are really interested in engaging with the botanical folks. Every 4 years or so, we have the Global Botanic Garden Congress. There will be one next year in Geneva, Switzerland, that's put on by BGCI. The Crop Trust, a wonderful organization we heard about yesterday. In the US, the American Public Gardens Association does have a plant collections network with some agronomic ties. I'm co-chair, actually, of the Agricultural Committee on that group. And then also the Plant List is a really great, relatively new website where you can put in any taxa, and it'll link to a whole bunch of other things. It'll let you know if there's NCBI sequence data on it. It'll link you to botanical manuscripts and all kinds of other natural history clearinghouse information. So with that, I also want to plug, a bunch of us wrote an article about how important botanical gardens are in the future of food. It was published last year in Nature Plants, and so I'd be happy if anybody wants to contact me or grab me. I'll be very happy to provide this to you by email. Thank you very much.
Now we're going to have our panel discussion. So if Rod and Ari could come up here to the table, as well as the other people that were invited to this panel session. And if the people who didn't speak already could just quickly introduce themselves to the group. We'll start with Savi.
My name is Sabi Natarajan. I'm a scientist in soybean genomics and improvement laboratory in U.S. Department of Agriculture in Bellsville. My research focus is assessing the biosafety of genetically modified soybean seeds, specifically the variation of allergens and anti-nutritional proteins, and both in genetically modified and non-modified soybean seeds, and to report what are the variations between those types.
So I'm Patricia Mergen. I'm working both for the Museum for Central Africa and the Botanical Garden in Mazen. I act a bit as liaison officer between the two and also with some funding bodies. And we have their experience of the Botanical Garden, as I showed yesterday, with many varieties of edible plants and mushrooms. And at the Africa Museum, we were involved in several projects on edible insects, which can also be interesting for this discussion.
To start off, I had one quick question that's sort of a combination for Ari and Rod. Ari, you talked about sort of data mining, collection specimens for learning new things about interactions. Rod talked about mining biodiversity literature for a lot of those same types of connections. We're sitting under a couple million volumes of agriculture literature. Are there benefits and other types of relationships that we can learn by sort of commingling the biodiversity literature, specimens that are coming out of traditional herbaria, the type of collections that FDA mentioned, and biodiversity literature?
Yes. So I guess many of the issues strike me as being fundamentally the same, and we are commingling the agricultural literature because huge amounts of stuff that's been scanned by the USDA is busy coming into BHL, which initially freaked me out. There are perhaps some differences. It occurs to me that one of the big chunks of literature which has been scanned by the USDA and come to BHL is fundamentally economic. It's price lists and seed catalogs. So I wonder, somebody must be able to do a really quite interesting analysis of the economics and the value of different kind of crops. I mean, you could go back in time. What was the price? Why did the price change? Is that purely market driven? Or is there some ecological or disease sort of response that we're tracking there? Are we seeing, are there some crops that, or seeds that declined in desirability for some reason? And we have a quantitative measure there, we have price. So you could, I guess, treat it purely as an exercise in economics and try and understand why those things varied. I guess we don't, in the sort of pure biodiversity world, I guess we don't really have much that's in a way that's quantitative like that. It's incredibly hard to value biodiversity in that way. Whereas if you can put a market price on it, then suddenly things become comparable in an interesting kind of way.
So yeah, the short answer to that question is yes. I'll get on my soapbox for a second. I think there's a deeper problem. I mean, the siloing has really become intense. My favorite example is I'm really trained as a population biologist and geneticist, and I was at a conference many years ago, and a graduate student from California had found a really interesting pattern. She was studying weedy perennial ryegrass. And every year she was going out and assaying these with a battery of genetic markers and finding just this new genetic swarms coming through. And she was coming up with all these evolutionary hypotheses of what was going on. And she was a student at University of California at Davis, which is simultaneously both one of the premier ecological universities in the United States and one of the premier agricultural and agronomic universities in the United States. Well, I'd worked in a turfgrass laboratory for a little while. And I said to her, well, you know, perennial ryegrass is reseeded every year on California golf courses. That's just their management practice. And they change varieties, you know, based on what varieties are good, and you know, there's constantly breeding going on. Where are your sites? Oh, yeah, it's funny. I collect a lot near golf courses. And she was one building away from the turfgrass department, at UC Davis, which is a really strong turfgrass department. And there was just it never occurred to her or any of her advisors or anybody else that she was in contact with in the ecology department to stop in and talk to the people who agronomically study the same species that she was working on ecologically. And so, what was a really well done biodiversity experiment based on the assumption that what she was going to be seeing out in nature was the result of natural processes was in fact an artifact of an agronomic process. And that's unfortunately quite indicative of the way education, at least in the United States, is happening, that there's so much siloing, and that's amongst plant scientists, right? Two branches of plant science. So at a very early phase, we really need to get in and start telling people, you got to talk to everybody. You can't have any more of this attitude of, they're an agronomist and I'm an ecologist and I'm a biogeochemist and they're a soil scientist or anything like that. We really need to get together and talk. At the very, very high levels, of course it happens. If you go into USAID, if you go to USDA, you go to IPBAS, you go to Crop Trust, UNFAO, people have figured it out by the time they turn 50 and end up in an important position in an international organization. But it's taking too long, and young scientists are really not aware of the interconnectedness of their areas, let alone the importance of the various collections that are going to impact them.
I think maybe the lack of collaborations with the scientists, it improves the connections. What do you think?
Change.
Yeah.
Oh, actually, in Belgium, we have the archives of the ENIAC, which is the National Institute of Agriculture of the Congo, during the colonial time. And when it came back independently, all these archives were distributed in Belgium in all kinds of institutions. Some were almost destroyed, lost, not well kept. And a colleague of mine tried for years to get them at least together, scanned metadata and so on. And she had the same issues that the scientists didn't buy in because they didn't see the added value on doing data mining on get this scanned. But finally, we find out that it also has a lot of meteorological data. And so now we submitted it not in the agriculture, but in the climate change, saying that we will mine data from that to make modeling on climate change, and suddenly we got it funded, or it will probably. So, but the thing is, it will become now available. It will be scanned. It will be OCR'd, crowdsourced, everything, and then the data for the agriculture also becomes available. And then we hope that we can also show its importance for the agricultural part. But the dialogue is really sometimes complicated, both with the funding bodies and finding the scientists who want to be multidisciplinary and saying, this is not morphology, this is not taxonomy, so we're not going to do it. And well, in my experience now working in both worlds, zoology and botany, because I'm with two institutions, I must say for my friends botanists that they are much more open with agriculture and so on, while in the zoology in general, not for all of them, but for many, I sense more that they want to stick to more classical science, and I have much harder time to make them come into such projects.
So one thing which struck me about your example of the golf course was it was kind of resolving that situation required that you had two people, I guess, talking to each other that just happened to be more or less in the same place. And so I guess my question is, what happens if that's not the case? So UC Davis, incredibly lucky, world-class ecologist, and you've got the turf people. But in many cases, that won't be the situation. So I guess one model, I think one model we sort of have in science is you make these kind of peer connections, you build up this kind of network of collaborators, which I think works extremely well if you're fortunate to have those collaborators in place. What do you do in a situation where, say for example, the people who knew about the golf turf seeding were geographically nowhere near the person doing the genetic study? The chances of that sort of being resolved would be very, very low. So I just wonder whether, if both those organizations were sharing data, and you could imagine that somebody was coming along at a higher level, like a global level, and aggregating all that, might that be a way to discover that?
Well, the story was that actually she didn't learn from the people at UC Davis. She learned it from me, and I was in New Jersey at the time. So we were on two coasts, and it happened at a meeting. And I saw her poster, and I said, "This is interesting. Have you talked to turfgrass people?" So, I mean, meetings are clearly important for that to happen, and that can happen. But the reality is there aren't a whole lot of people that know turfgrass management at an Ecological Society of America meeting. So yeah, I mean, the point is still really well taken that there are these areas of knowledge that are still not easily accessed, especially management practices. And I'm not aware of any really good clearinghouses for management practices for at least not for the big agronomic crop species. I can tell you at Botanical Gardens, we really struggle with this. This is a topic on the international level. We keep pretty good records of the plants we have, of the seeds we have, and things like that, but not how we grow them. And I'm only aware of one or two gardens, both in Europe, that have developed in their collections database software areas where the gardeners who take care of the living collections have to put it in information. I fertilized this plant on this date, so that you can go and mine the data. Maybe you have, many gardens have trees that are hundreds of years old, or other species that have been around for a long, long time. And you need to know how somebody 50, 60, 80 years ago took care of that plant, and look at, are your environmental conditions changing? Is that going to change if you want to have lasting living collections? We don't capture that data, although some are trying to. So that's an example of what you're talking about. There are all these examples, and we really are, I think, falling down in many, many ways. I shouldn't say falling down. I have a lot of optimism in humanity, but we're missing a lot of efficiencies because we're letting the scarcity of resources prevent us from being more comprehensive about our data gathering and communication collaboration.
How to improve the efficiency? Okay, I'm sorry. How to improve the efficiency? I'm just asking. So how to? I mean, we know we have a lot of problems, and then we don't have connections that much. How to improve the efficiency?
I think informatics is probably the best possible way, and then the bottleneck becomes getting the data into an even researchable system, and connected to systems where people are going to look. I think you know more about that than I do. But it's hard to train people. I have a workforce, and they're struggling to take care of the plants that they have. You give them an iPad and tell them, okay, now after everything you do with this plant, throw it into the iPad in a way that uploads it to some database so that becomes searchable information and shareable. It's a whole new way of doing business, and I think that comes from education. So a good breeder, you don't go anywhere and do anything without your log, right? You're always noting what you see that's interesting, and not all disciplines are so well disciplined in that area. And then the second step, of course, is getting it into a searchable, shareable information format.
Building a little bit on what Patricia was saying in the difference between zoology and botany, Botanic Gardens in general have done really good jobs with things like Ari was showing in terms of the wheat and sort of getting that information out to a wider audience, whether it's something very common like wheat or more exotic like vanilla, where you can sort of see where that comes from. Zoology doesn't quite have that venue for showing that. You have zoos, which are much different. And even sort of like our own National Zoo recently closed its farm exhibit, where you could actually see agricultural livestock type of thing. Those of us live in places like Arlington County here don't have state fairs where you can see massive amounts of big livestock. So Patricia, I was just wondering if there's any other sort of venues where you can see, for instance, your own institution mingling or providing an output reach for zoologists to have that kind of actions.
Well, I was in, for example, I think it was in Stuttgart, where the zoo is actually really close to the Natural History Museum. It's just next door. and they work together much more closely. And I could see a different attitude there, because when an animal dies there, they get it immediately, and there's more connections also to preserve samples and so on. But we discussed it at the Africa Museum, and they directly said, no, this is for the Zoo of Antwerpen. So we have connections with them also. We try, if an animal dies, to get it if we don't have too many already. We had the Wildlife of Barcode project a bit, where we managed to have agreements with zoos that they give us fresh samples to analyze them genetically. So we try to go there. But in general, there is more this vision that Natural History Museum are for, in zoology, for dead animals, while in the plants you have the living collection and the greenhouses, which is more or less their zoo, which is completely embedded with the dry collections, while in zoology, only the visitors sometimes come and ask me, where are the lions? And they look everywhere, and so sometimes the public doesn't understand that we have only dead animals. But the only thing I can see is to have closer collaboration with, for example, this project with zoos, and try to change things a bit following what botanists are doing. Now, we had exhibitions on spiders, live spiders, and we had a lot of people coming. They were really happy. So several museums have permanently also living insects or herpetology, and that attracts a lot of people and also does a bit of change of mind. We had a project on edible insects where we tried tasting evenings and night at the museum and things like that, which has also quite success. But there we run into funding issues, because the funding body and the European Commission, they don't want to hear that we explore all the species like we did in the smaller funded project. They really wanted something with 2 species or 3 that works directly, and you can have proteins for the European market that they can sell and make money out of it. And the work package we wanted to do in that European project with taxonomy and exploring in Africa all the possible food sources was not accepted by the others, because they said, we have no time, we want money now, and that it works in Europe to feed and to have the market, and in other places fight against poverty to have proteins. And they said, you are coming too late, and we're not interested to know about all these species you have. We just want 2 that we know they are healthy, no side effects, and that we can have quickly to the size that we can sell them. And so we didn't join the project because they didn't see a place for us.
So I guess it occurs to me that, so for example, when we talk about the literature, so we can make a case for literature being of enormous value. And I think it is, because I have invested a lot of time in trying to find that value. I guess for an audience like this, I guess I have a question in a sense, is that if your focus is, say, more applied, does the literature have that value? If you're an academic interested in the study of biodiversity, it's a no-brainer. It's where a huge amount of information comes from. Is it just a sales pitch to say that, well, there must be useful information there about agriculture and crops and food and diseases, or is that just something we spin to make it saleable to grant agencies? I'd be generally interested if people think there is a valid case that the scientific literature is, data mining it would be of use, or is it just something that seems a really kind of cool thing to do? We have somebody running.
Hi, so Maxine Levin, the U.S. Soil Survey. This is something we've been wanting for years and years with the Soil Survey. Over 20 years ago, we made a recommendation that, at least amongst our collaborators, that we would ask for a centroid point to identify where their research had occurred on soils and then add it to the soil survey database, that we would have a layer of content management that would be place-based. So let's say if you did an area of interest somewhere in Nebraska, you could also search to see what research had been done specifically on that soil or in that region, that then you could data mine. And to believe it or not, we still don't have it within the national database for soils. Even a recommendation so simple as just adding a centroid point to every piece of research. And I wonder what the mechanism would be to make that happen. It just drives me crazy. And it's so important, particularly in place-based lines of research, such as soils or agronomy, where really it's place-based as the number one line of thought. And I wrote down everything you had written on about, the BioStore databases and the GBIF database, because, that's the key, I think, to a lot of agronomic research and collaboration within the U.S. I just wonder, how do you get the computer scientists and the GIS people and the folks who collect literature and put it into the database to insist upon a centroid point for every piece of literature and research that comes about? Like an input, like for when they log their research in to a database, that on the form it says you have to put a centroid point that identifies your research as a place-based piece. Very important for agronomic research.
I think we had a question back here first.
The big data in the literature is all fine and dandy, but nomenclature has changed over the years. And I'm going to use two very dirty words. The lumpers and splitters have been added. And there are multiple cases where in the past, like with Echinacea, originally there were two species. They got lumped into one. They got split again, but the names were reassigned in reverse. So now if you go back to the literature prior to 1920, what is one species is another species. And yet when you do searching in databases, all of a sudden you're going to be led down a Mary Chase going in the wrong direction with some of these. There's also other issues. I find it a lot in the nomenclature of cultivated plants, that there's a lot of confusion, especially then when you start relating cultivated plants to common names, i.e., you had the exhibit of the wheat. What's spelt? Is spelt a wheat. It becomes very important from a regulatory point of view because wheats are allergenic. We have people maintaining spelt is not wheat, so they don't have to label it as an allergen. But again, you have to go through the literature. And sometimes it's very confusing, and it's amazing what lawyers come up with. So how can you correct for these nomenclature errors that are propagated in the literature? And what control mechanisms can you have for them?
Anyone want to answer George's question before the next one?
Yeah. So I guess this is the taxonomic concept issue. So there will be cases where names aren't enough. Names do change. The simple cases are where people basically mess about, move species between different genera, and they're relatively straightforward to track. It can be slightly tedious. The really messy ones are the ones you point out, where the name itself, the meaning of that name changes dramatically. There are people who develop various kind of markup schemes to sort of flag, when I'm using this name, I'm using it in this particular kind of context. And so I guess one way you could imagine solving that would be, if you have that literature that's relevant, you'd go through and mark up, okay, this paper means this name, this context, and this one's the same, and this one is a completely different thing. And if that markup were stored as some sort of annotation, I guess ideally you'd have that resolved for that one case for all time. So you could imagine a situation where BHL would tell you not only this page has this name on it, but actually when you see that name in this paper, it means this, sent to somebody. And later on, you see the same name actually means something different. I'm going to go out on a limb and say I think these will be edge cases. There will be a few of these cases which will be really annoying and quite tricky. Most of them I don't think will be that hard, but there'll be a few that are really, really messy. Can I just come back to the soils? So I'm slightly confused. So I would have thought that people doing research today would be walking around with GPS devices or mobile phones and would record stuff. And in which case, I guess you need to encourage people to do that and maybe give them brownie points or a star or something. A lot of the dots on maps, for example, you see with GBIF, with biological specimens, that's reverse engineered. People have recorded place names like 5 miles down the road. And there's a whole software industry of converting that into a point with some degree of uncertainty. So this is georeferencing, and the vast majority of data in GBIF is inferred in that way. It's not actually from GPS devices. The modern stuff is, because people care greatly about it. Not as many people as David would like. There's a whole industry of papers complaining about biologists who don't record that information, but it's becoming much more common. But a lot of it is done retroactively. So if you have that data, if you have some sort of place information, there are software tools that will help you convert that into a dot on a map.
Maybe to answer the question on nomenclature of cultivated plants and how you find the common names in the literature. So I would say for wild taxa, it's already a challenge. Also with multilinguals, but with cultivated plants, you also have the issue that there are many and there has hybridations. And I have a colleague who just started a PhD on pelargonium, so these geraniums, the plants, and he said it's sometimes real nightmare because they hybridize, and if he tries to do genetics on it, it's really difficult even to put a scientific name on it and to find out how they hybridized, how many species there were, and all that. And I saw when we went through the living collections at the Botanical Garden, we have many. We know it's pelargonium, but we only have the genus name, and we need to do genetics on them to put the variety and so on it. So, and breeders keep doing new cultivated plants to sell them and so on. So it's difficult to keep up. And then if you look in the old literature, to link common names to the scientific name of cultivated plant is really a challenge. But I think it's possible, but to my experience, there is a higher rate of error than with wild relatives.
Hi, I'm Lisa Castleberry over here, the research mycologist with the USDA Agricultural Research Service, and also currently the director of the U.S. National Fungus Collections. And I just want to get back to Rod's sort of original question of is it worthwhile to record these kinds of data? Our laboratory and the collections has a long history of recording exactly that kind of data. We have, I don't know, I think Shannon, our collections manager, could answer this a little bit better than me, but we have on the order of 400,000 to 500,000 records of plants and the fungi associated with them and the location in which they were found. Now, we don't have GPS data for a lot of these, but we have a state in the US or a country or somewhere around the world. And we have on the order of 300 unique visitors per day to our website searching exactly that sort of information. And this is in addition to our specimen data, which records, all of the approximately 900,000 specimens that we have in the collection, which is another vast resource of this sort of data. The majority of our users are regulatory agencies. So the USDA's Animal and Plant Health Inspection Service is a tremendously large user of our data, and that's meant, to help curtail issues of invasive species, emerging pathogen issues, and these sorts of things. This is tremendous economic value in that, just in terms of imports and exports on a daily basis. So I think that yes, just from that standpoint alone, it's tremendously important and valuable. We fund this. We do this. I'm very intrigued by the OCR method and the data mining, because we employ 2 people right now to search the taxonomic and agricultural literature for new reports of fungi on particular plants, as well as, to George's point, on nomenclature of agriculturally important fungi, which has its own nightmare of issues involved in it right now due to some changes in the code of nomenclature for names of fungi. So we're doing this manually right now, and the explosion of literature these days has made this much more difficult. We certainly utilize Google Scholar for alerts as to new reports of literature for these things, but it's hard to keep up. But as to the point of, is it important and is it valuable? It's tremendously valuable to agriculture, and certainly regulatory agencies recognize that.
Can I just quickly follow up while the mic is moving around? So a lot of that could be automated in principle. In fact, there was a neat tool developed, about 10 years ago, for monitoring feeds from, say, PubMed and other sources and looking for new species names. So you find a species name with NSPA or SPA, NOV, it's a new species name, and you got an alert. So I guess what you'd want is a tool which can find scientific names in literature that exists. And I'm guessing there's probably only about 5 or 6 different ways people write new species of fungus found on plant X. And if you've got a script which can do that, then you could automate that mining. Have you heard of the Global Interactions? Is it the Global Interactions database?
Global Biotic Interactions.
Hmm?
Global Biotic Interactions, globally.
That's it. So it's sort of loosely attached to Encyclopedia of Life. I think I've got that right. So this is a database where people are basically trying to assemble a global database of interactions. Who's on what and where. The database that you've got, would you share that with that? Yeah. Right. That would probably be okay. I mean, basically, they will take kind of Excel spreadsheets, dumps, whatever, more or less, and play with it. But I can imagine that would be an amazing kind of thing to combine with the other kind of interactions data that people are pumping out.
So following on that, I want to say that here at the library, we are building that sort of expertise. And later this afternoon, I'll be talking about some of these activities that we're doing here in the library and how they relate both to sort of the museum informatics world and the agricultural informatics world. And what always strikes me as interesting is that mining the legacy literature makes a lot of sense. There's no other way to get these structured pieces of information out. It's done and gone. What boggles my mind is that going forward, this explosion of literature is still subject to these same problems. And I'm hoping that the situation will change now that publication is moving towards requiring data to be published along with their papers. And so the challenge is to raise the bar for how those data are managed going forward so that people know that they should tag it with a lat long, for example. Now, in the research community, as people have mentioned, there's all sorts of activities to mine, to pull out locations from text, that sort of thing. So where we need to go is get the tools that are being developed by the research community into these government production level systems to, for example, suggest to an author who's archiving their data, do you want to put a pin on the map right here? So rather than ask them to figure out the lat long if they didn't have a GPS, who knows, use these as suggestion systems that they can then confirm, yes, this is the metadata for my my study. So I don't think we're there yet, and that's basically what my talk is going to be like this afternoon. But a lot of the pieces are there. We just have to somehow get everything linked together and put those tools into place in these long-term archival systems.
David, and then
Thanks for two great presentations. I want to try to crosswalk some issues that you raised. Big data long tail about literature, lots of small articles about many, many species, but very few of them are edible. Very few species are edible. And I think yesterday when Grace was talking about BHL, their efforts to digitize more of the agronomy literature, I think that may change, that you're going to find in those very common species occurrences in the literature, you're going to find more and more literature about those few plants. Ari's point that herbaria are not digitized, I think that's the taxonomist's view, right? We need to figure out all the names. We need to digitize all the sheets. Yet at the same time, you recognize there's only, 1,200 edible plant species. You have exhibits on 12,000. But we're using very few species. And you've devoted space to really interesting exhibits on them. Why aren't we focusing on digitizing just the ones that are of economic importance and try to make those records richer with links to the literature, with all of the species, the data richness that I spoke about yesterday? Rod, to your point, yes, it's true. You can navigate from name to literature to GenBank record to locality. You can start anywhere in that tetrahedron, and you've seen my diagram about the tetrahedron of traits, literature, name, specimen. You can navigate among them, but a plant breeder who's interested in finding collections from an area with certain traits where there are background publications they can look at, that's not a search you can do. Is there a technology, is there an informatics trick that allows us to say, find me the intersection of these 4 characters in those 4 different domains? Specimens that, for example, are fit for use, that have preserved tissue that can provide DNA, that have been published in the literature, that have localities in my area of interest, that have the following traits. That's the golden fleece, I think, for making academic botany and applied botany real partners. The other missing ingredient, the motivation. Why go to all this effort? I think there's a very small subset of institutions that have the vision, their leadership has the vision to say, this is the key to sustainability of what we do. Social sustainability, economic sustainability, operational sustainability. We have to increase the pool of stakeholders and our public awareness, the way you're doing in the Botanical Garden. And by the way, I did find the Nature Plants article. It wasn't open access, but since you're a Fed, I found it on the Botanical Garden site. No, the law says you cannot charge for you're a public servant, right? Yeah, well, it's out there on Twitter. It's out there with a shortened URL to the full text. So yeah, I think putting your two talks together, you put your finger on where huge opportunities are. Question is how to crosswalk some of the challenges and some of the commonalities between what you've said. By the way, the GPS issue, that's been raised in the literature. Why are there so few GenBank records with GPS localities? Very small. We did a survey of them. My other job besides Cycle is head of the DNA barcoding consortium. Virtually 100% of the DNA barcode records in GenBank have GPS coordinates, and that's because we have a data standard. Fitness for use as a DNA barcode record includes a locality and a link to a specimen in a repository. That's fitness for use for us.
Any comments on David?
Yeah, before I ask my question, you want to give them a chance to answer.
Yeah, so the query itself is trivial, quote unquote. We could actually, in principle, do that. The part of the issue is having all the data and having it all connected. So locality is trivial, lets and longs. Traits, EOL is doing quite a bit of work on traits. You'd need to have the traits coded in such a way that you and I would do it and would agree. So some gross things, I guess, like C4 photosynthesis or something like that, we could probably agree on some sort of system. We don't have all that kind of data in one place that we can do the query on, but the actual query itself is essentially trivial. In terms of why we're not there, if you look at the communities that are there, there's an incredibly strong driver to link stuff together, and typically it comes down to money. So we have links between literature and people and funders and organizations because there's money at stake. The trick to get to the promised land is to figure out what's the value that'll drive people to make those links. So one approach might be, if you're an institution, you want to be able to get money. How do you demonstrate money? You demonstrate your collections being used. That's a link between specimens and literature. So you have a commercial interest, in a sense, crudely put, in showing that. That may drive that. And that's something we're kind of interested in at GBIF, is trying to link those things together. But there needs to be, beyond the academic, isn't it cool, which is my motivation, you need to find somebody who basically would put money or attribute strong value to those links. Otherwise, we're just messing about.
Those are for the breeding center, not for botanical gardens or area. That's not what they're targeting. And just to be clear, I wasn't even saying search on the value of the trait, just search on the fact that there's trait data.
So I mean, I think there's two ways to address that. One is that at least for botanical institutions, we're still coming out of a phase of, sort of pure research, and rediscovering that connection with economic botany, although the Society for Economic Botany still doesn't consider agriculture economic botany, which vexes me to no end. But we're coming out of it, and there's a very strong willingness to do so. So I think the time is nigh, and minds are fertile and ripe for this kind of conversation. But you do have to follow the money, and sometimes the money isn't the direct thing you follow, but the regulatory, that you mentioned before. So, one of the best reviews of primary, secondary, and tertiary gene pools for a wide variety of crops, going well beyond the kind of big economic crops, was undertaken by, I think it was the EU, published maybe in 2008 or 9, really based on introgression concerns around genetically modified agriculture. So, sort of getting into that whole conversation of, well, if we do modify X crop, where might we want to avoid planting it because there's some chance that we can quantify that it might get into a related gene pool. And whether or not that's a bad thing is they put aside. We just said, what's the chance of introgression? So there's a 400-page book out, and all the data in terms of where the geographic locations for those gene pools was mined from Botanic Garden or from Herbaria, but the information about the gene pools was mined from agronomic literature, because those are the people who were seeing what was cross-compatible and breeding and how often and with what difficulty. So where it's useful for a moneyed interest, and the regulatory folks are going to follow where there's potential industry or existing industry, it does happen, and it happens at very high quality. And so I think, the question then, your earlier question of, should we target, digitizing the 25,000 species? That's an interesting point. I'm not aware of any herbaria that are specifically, usually they say types first and then kind of everything else. So, I mean, those are interesting points to bring up, but making the connections, the money does follow where you can show that there's really economic, meaningful.
Even in 12,000 economically important plants, I don't think it's 12,000, right? I mean, if you... Oh, okay, because, I mean, if it is less than that, you... Okay.
Well, what we also identified is the query to interlink actually the data. It's doable, but as Rod said, the critical point is really to have this data into the system and in the right format. And we did estimations, I think, a bit everywhere, how much it would cost to fill that in with own stuff, be it recent data that you collect doing field expeditions or data mining from old literature. And actually, the thing is going to crowdsourcing and citizen sciences. But it's also not that this doesn't cost anything, because you need still to set up the system and find incentive that the people do it. And then there's also a big debate. While there were statistics done that the data collected by crowdsourcing and citizen science are almost as good as the one done by scientists, but others don't agree with that, and they say it takes them as much work to control the data than have it filling in by third parties. I have fellow scientists who are really afraid that showing that it's possible via crowdsourcing and citizen science, that this will say to the funding bodies, we don't need you anymore, so they will lose their job, so they will not be renewed. And on the other hand, to set up citizen science observatories in developing countries is really challenging. There are trials, there are also calls in the EU now for in situ observatories for agriculture in Africa. But by tradition, we are used to pay them a per diem for any work we ask. And so when we now go and we try to set up projects, many don't understand, the local farmers, that you will ask them to go there to take soil samples, to look at water quality and send all this information, and you don't give them any money. You just say, do it with citizen science or crowdsourcing. So there have been some crowdsourcing citizen science events in Africa. It's working. We have something called Jungle Rhythm to collect threads from forests. That worked, was filled in quite well. But there's still a lot of discussions because people expect to be paid for this kind of job. And there is really the issue, especially if it's more work than just transcribing.
Okay. It took me a little while to get the mic, so I'm going to maybe take us back in time a little bit to an earlier question about the quality of the data. And I had this question yesterday when we had talks about other databases. As we build these really large databases that are immediately put out into the public or not, I guess, and scientists and people are using them to do these data mining projects, the quality of the data comes into mind. And maybe we think that there's so much data that'll override the bad quality. But even so, it seems to me that there needs to be mechanisms for correcting mistakes. So if the taxonomy changes, or if somebody does a data mining project and learns that this whole data set had their locations wrong or something like that. I mean, it may be one specimen, it may be a problem within the database. Do you have mechanisms for corrections to be made, including corrections that are found from people on the outside? And if you make corrections, then there seems to be a need for version control so that a person knows, when I did this study, I used this version of the data set, because whatever we do now, we think that we're doing it better than it was in the past. But 10 years from now, 20 years from now, they're going to do it better than we do now, and corrections are going to be needed. What mechanisms are there for doing that?
So I mentioned the BGCI, Botanic Garden Conservation International, which maintains the aggregate data of living collections in botanic gardens worldwide. And the system, this is just an example, but the system there is each institution pushes them data typically once a year. And so as each institution are going through our collections, we're accessioning, deaccessioning, looking through everything, updating taxonomy. If we come across, if we find, records of provenance data or, anything else that we can add into the metadata, we're doing that. We're pushing that to them once a year. And then once they get that push, it's in their system and it's queryable through their system. And so then what they do is there's a pretty basic search that you can do through them as an outside researcher. And they ask you to, if you use that, just, state specifically the date on which, that should be in your publication date, the date that you did it. But there are also much, much more advanced data capabilities, in which case you have to work directly with their bioinformaticians. And then the same thing, whatever you want that they're going to create for you, they're going to provide that and give you specific instructions. We compiled this data on this date, please cite in this exact format. And what I don't know is if they save yearly versions of the data. I know we do as an institution. We save our collections data yearly so that people can go back through time and see what's done. And there's also annotated ways of doing that other than just having full. But I think it's a pretty, somewhat standard for strong data curation folks to have mechanisms to deal with these kinds of issues.
So my take on this is the only way to discover data quality is to use data. If the data is not used and not visible, you're never going to know. So think in the context of something like GBIF, which has something like 600 million records. Often people will send data to GBIF that is crap. And you don't see that until you put it in global context. And suddenly you'll see all kinds of patterns, which are just clearly artifacts. So, we talk often about peer-reviewed data. I think that's a myth. I think the only way you peer review data is if you use it. So you've got to expose it, you've got to use it. There is this model which has basically failed so far, this notion that there's all kinds of issues behind this. But for example, one model is I'm a data provider, I provide data. If somebody spots an error, I'd like to be told and I will fix it and I'll give you back the clean data. By and large, that doesn't work because it assumes that the data providers have the resources to fix the data. And 9 times out of 10, there's nobody home, there's nobody who can do that. Quite often what happens is the mission is get your data into GBIF, you do that, you tick box, job done. There's nobody who can actually deal with record. Now, there are some big institutions which do have staff which can do that, but by and large, that's not the case. The model that I would favor is very much the way we handle source code. Open source code, the code is out there, anybody can have at it, debug it, fix it. You have versions, you have version control, you can see exactly what happened. I would love to see the same for data. Data should be out there, it should be treated as something which the community can edit, work with. And if you're a software developer, all this is being solved 30 years ago. We have version control, we have all this kind of wonderful stuff. We haven't adopted that mindset for data, partly because people are incredibly possessive and people have all sorts of issues with ownership. So there is this model that many people providing data would like to be told directly about the errors, which is fine. Unfortunately, very few of them have any realistic means of fixing it. So I'm in favor of get the data out, get it out quickly. The more people you expose it to, the more bugs you'll find. There's a saying in software that all bugs, given many eyes, all bugs are shallow. I can look at GBIF, I don't need to know what the organism is, I can tell you if something's wrong. And then you need some mechanism where the data can be edited and cleaned in public so we all get the best quality data we can get.
Well, to say
annotation, I guess some form of annotation is going to happen. But again, the notion that you wait for the provider, I mean, that's the hang up with GenBank. GenBank has this bizarre system where, if you're a postdoc and get hit by a bus, or you go and do something else, and you're the person who submitted the sequence, GenBank won't fix it. I understand. So, it's an issue.
Well, I would like to second that it's really the issue with the orphan data. So often these projects for putting data online are on soft money, or sometimes you have big projects, and if they are still active and used, and you have a good annotation system, you can have regular updates. But as soon as the data owner or data provider is not there anymore, you have an issue because you don't know who's the authority to change it. And there, it's something we need to work on. For example, for the Catalog of Life, they have the Global Species Database. They also have some who are not maintained anymore, but they try for each bigger group of taxa to have a few people responsible for this taxa, and that if there is something, they can somehow decide if it's updated or changed or not, even if the original provider is gone. But it's difficult to set in place because of this ownership requirements. Yeah. Yes, yes.
So, in theory, you could submit a new GenBank record with the same sequence of correct identification.
Yes.
So now you have confidence that you have both users
Mm-hmm.
and have to achieve it. Only the institution can submit something from that institution.
Yes, but I'd like to change that. But I guess there are 2 points here. One, with GenBank, they have a rather bizarre system. The beauty with GenBank, though, is errors are discoverable. You build a tree, something's in the wrong place. So, yes, there are errors, but they're really relatively easy to spot. With other systems, you're not so lucky. There are lots of people thinking about this general issue of annotation. There's a system called Hypothesis, hypothesis.is, which is coming out, which you can probably see on most journal websites probably in the next couple of years, which allows people to go and annotate text and annotate databases. So this is an issue that's sort of much bigger than sort of biodiversity agricultural community. So there will be, I think in the near future, a mechanism where people can add comments and specifically challenge statements that are made in databases or on online literature. It's free, it's open, and that's very, very cool.
I'm afraid we're going to have to skip Kevin's question. I want to give a round of applause for everyone on the panel. And you can continue the discussions during our break time, and we'll resume at 11-ish.
Okay, everyone, we're going to get started again. All right, so I am pleased with the way the discussion went before, so hopefully you're ready for a couple more rounds of those, of the panel discussions and group discussions. Our next session is going to be chaired by Rosalind, so come on up and we'll get started.
Good morning. My name is Rosalind James, and I'm with the U.S. Agricultural Research Service. I'm National Program Leader for Invasive Pests of Crops. And I'll be chairing this session this morning until lunchtime. The first speaker is Kevin McCluskey. We heard him yesterday with Kansas State University.
Thank you very much for the opportunity to share a little bit about the collection of which I'm the curator, and a little bit about what we're doing with the resources in that collection. And I'm going to go down a few levels in resolution. So we've been talking about taxonomic issues and about global issues, and I'm going to talk about genetics. And the comment I was going to make earlier was that we have an opportunity to benefit as a community from what's being done with personalized medicine, where every human could have a genome. What we're doing at the Fungal Genetic Stock Center has absolutely benefited by the Department of Energy and the Human Genome Project. Now genome sequencing makes it possible to even consider the idea that every strain in a culture collection could be associated with a full genome sequence. So the Fungal Genetic Stock Center was established in 1960 when there was a concern that the materials, the research materials that had been used in the seminal demonstration of the one gene, one enzyme hypothesis would no longer be available to future generations of researchers. It was established at Dartmouth College, and the FGSC has been rather peripatetic. It's moved 4 times. The first 3 times were associated with retirements of the collection director. So I'm the 3rd curator during these 50 years. In 2012, so the FGSC was supported continuously by the NSF, originally through the Living Stock Collection Program, and more recently through the Collections in Support of Biological Research Program. In 2012, the NSF changed their funding, and they're no longer supporting what they call sustenance. So that would mean, salaries for curators, such as myself. Rather, they've looked to project-based 3-year grants with smaller funding caps. And they merged with the Natural History Collection Panel. And I have to tell you that living collections are smaller in number. There's about maybe 20 living collections in the U.S. and maybe hundreds of natural history collections. Right now, the NSF program supports about 8 living collections and over 85 non-living or once-living collections. So this funding change impacts all the living stock collections that are through the NSF. We call it the stable of collections, And so this is what precipitated our move in 2014 from the University of Missouri, Kansas City, which was no longer able to offer us space and resource support when we didn't have an NSF grant. The Department of Plant Pathology at Kansas State University offered us a space and some salary support. And this was through a process where we issued a request for proposals distributed widely among universities in the U.S. 8 institutions expressed interest in supporting us, 2 institutions supported proposals, and K-State came through with salaries and space. So then we loaded up the collection into a truck, and I can tell you the Fungal Genetic Stock Center weighs 14,000 pounds. So what makes up those 14,000 pounds? We're a very different kind of collection. Biodiversity isn't our main emphasis. We're really in support of one major research area, that's filamentous fungal genetics. So we have over 20,000 isolates. And we have a number of smaller number of isolates of other fungi. Aspergillus was the second major research organism used in fungal genetics through the classical genetics era. And the organisms I've highlighted in bold there are plant-associated or plant pathogenic fungi. Neurospora is always associated with burned vegetation. That was very, very commonly collected in sugarcane plantations after burning. The biodiversity collection is mostly Neurospora, although we do have over 77 different species from 23 different genera. And of that, we have almost 60 species with fewer than 10 isolates. Now we have over 70 species where we have the reference genome strain. And what I'm going to talk about later will show you how the reference genome strain is just the first crack at it. You know, we have now 600 strains with full genome sequence in our collection. We also have molecular tools for working with these fungi, and these have allowed the collection to have impact in a diverse area, including the plant pathology and human pathology communities, because these plasmids and cosmids are often used in other areas of inquiry besides our main target community. The collection grows, I mentioned yesterday, kind of like a snowball rolling down a hill, you know, with Rocky and Bullwinkle. It just keeps getting bigger and bigger as people send you things, often when they retire. So there's two kinds of deposits. Those are individual deposits when someone wants to leverage the impact of their materials by making it publicly available. And then when somebody retires or when you have a large community resource. So you can see here the impact of the, is this point? Well, after around 2006, we started accessioning GMO strains of Neurospora crassa. Once the genome was sequenced, there was a whole genome gene disruption program. And so now we have a gene deletion mutant for almost every gene in the organism. And I say almost because through genome annotation, you get a lot of little pseudo genes or presumed genes or predicted genes. So we have about 3,000 wild type strains, and those are mostly Neurospora crassa. We have an additional collection of 4,000 wild type strains from the collection of David Perkins at Stanford University, and these were accessioned when he passed away. We got the entire collection. They also have geographic information and substrate information, but that's not part of the main collection. We also have GMO medical yeast fungi, so these are in arrayed sets that we distribute on behalf of the medical research community, and we have 49,000 strains of Magnaportha, which is a rice pathogen. These carry the tDNA from Agrobacterium. So this is a tragedy in one regard because we've never been able to distribute them. They require a special permit from the USDA Biotechnology Regulatory Service within the US, and their Magnaportha was developed by our government as an agent of biological warfare in the 70s because it's a rice pathogen, so we can't export Magnaportha. Actually, an individual at Delaware University has a permit now to obtain and use these materials almost 10 years after we received the collection. And so there's been over 500 individual depositors throughout the tenure of the FGSC collection. This just shows the kind of specimens that we maintain. We're very fortunate that most of these fungi can be freeze-dried or can be preserved by putting spores in milk on anhydrous silica gel. So you have a little slant that's shown on your right. Sorry, actually the silica gel slants are here with the caps. And so you sprinkle a few grains of that silica gel into a culture slant, into agar media in a test tube, and it grows up over a few days. We also have about 20,000 of these silica gel stocks, including the GMO strains. And for things that can't be preserved on silica gel, we have them in liquid nitrogen as glycerol stocks. So the major research system in the Fungal Genetics Stock Center, of course, is Neurospora crassa. I mentioned it was used to demonstrate that biochemical traits could be inherited as single characters in the 1940s. And for this, Beadle and Tatum won the Nobel Prize. This is considered the central paradigm of molecular genetics, the one gene, one enzyme hypothesis. Neurospora is very common in the environment. So what you see here is a pile of sugar cane tailings after the sugar extraction. And the orange color is all Neurospora. So you know, that's several people tall right there. It also turns out very recently we've been able to obtain similar pictures and isolates from sugar beet processing. So Neurospora is associated with sugar extraction globally. Because of its association with plant cellulosic material, it's been developed as a model recently for biomass deconstruction in collaboration with the US Department of Energy. And a recent demonstration showed that this plant association goes so far as Neurospora being potential, well, it's certainly an endophyte and potentially a pathogen, probably a pathogen. So one of the things I wanted to show you was that these historical strains, these genetically characterized strains, can gain new relevance in a modern era. So this shows the distribution, the number of requests we had from clients in our community for Neurospora crassus strains carrying the genetic marker OZ2, which encoded osmotic sensitivity. It wouldn't grow on 0.6 molar sodium chloride. So over many years, it was requested a few times as a genetic marker because Neurospora was a genetic system in the classical genetics era. In 1999, it was characterized, the OZ2 was characterized for its ability to confer resistance to phenylpyrrole fungicides. And overnight, as it were, this strain and strains carrying this marker became the most popular strains in the collection. The other thing we carry that's relevant to food security and plant pathogen research are VCG testers, so vegetative compatibility group. If you want to understand where your strains came from and some of their genetic characteristics, you can do this very straightforward. In a very straightforward manner, and also mating type testers. And these are important for standardization, so people all know that they're making the same comparison. So if you say it's mating type A, well, it's compared to a strain that everybody can get. So I keep mentioning that Neurospora is a genetic system. It had one of the most densely populated genetic maps in the classical genetics era, with about 1,200 markers. And I say about because subsequent analysis has shown many of these to be allelic, mutations at the same gene. Some of the markers are genetic duplication, so chromosome aberrations. Some of them are chromosome regions like the centromere or telomeres. Of these 1,200 genetic markers, only about 800 were linked to an open reading frame now that we have the genome sequence. So that means there's around 400 anonymous classical mutants. And what that means is there's a trait that you can follow in a cross. So it's a genetic system, and these traits segregate as a single locus. And what you can see here on the left is what the wild type colony looks like, and that's a 15 centimeter Petri dish. You inoculate Neurospora in the middle, and over the weekend it's grown out to the edges, it's conidiating profusely. If you leave it too long, it grows out of the plate and starts heading out across the bench looking for mating partners. And the other mutant, semicolonial 8, is just a morphological abnormality, and we don't know what the gene underlying that trait is. And this map just shows part of linkage group 1. It shows linkage group 1 and the different kinds of density of markers on linkage group 1. And these are all phenotypic markers. So I contrast that to molecular markers like amplified fragment length polymorphisms, which have been used more recently to make densely saturated maps in other organisms like Fusarium. So we've used a number of approaches to characterize these anonymous mutants in the Fungal Genetics Doc Center, and this was directly related to our NSF support. We weren't supposed to do research. We weren't supposed to just ride the coattails of the collection, but we were allowed to do research that added value to the collection. Identifying what a particular mutant was considered adding value. So what's shown here is a molecular approach to characterizing temperature-sensitive mutants. On the left, it says no DNA, and on the right, it says wild-type DNA. And what we've done, we've taken a temperature-sensitive mutant, and we've introduced cosmids from the whole genome sequencing program. We can walk using cosmids that overlap from a known marker towards the unknown marker. And when you grow these at 37 degrees, if you've complemented it with the wild-type gene, it will grow at 37, and if you haven't, it doesn't grow. And these are pretty tightly regulated. Another thing we've been able to do is to do targeted sequencing of known ORFs. Here we go. And this was a project that we actually did with high school students. So we all know what the TRP3 mutant locus is. It's been pretty well characterized. It was very heavily characterized in the classical genetics era. And on the line in the bottom here, it shows the classical genetic mutants in the black text, and then in the blue boxes, the blue triangles show where we found a single nucleotide polymorphism mutant. The red hexagons show where we found stop codons, nonsense mutations. And this was done by high school students who came in and used the polymerase chain reaction. I should add the polymerase chain reaction was discovered by scientists who got a strain from a culture collection. And the green thing is kind of cool because in the original research in the 1970s, those mutants were shown to be immunologically non-reactive. And what our high school students showed is that the non-reactive ones weren't making proteins. They had nonsense mutations. They had a premature stop codon. So that's kind of laborious. The cosmid walking, well, you can only get to certain kinds of mutants. And the targeted sequencing only looks at things that you already know. So in collaboration with the Department of Energy Joint Genome Institute, we undertook a pilot study to use whole genome sequence analysis to identify otherwise anonymous mutants in the Fungal Genetic Stock Center collection. And so this shows a mutant called Compact. I keep wanting to point. Here we go. This shows a mutant called Compact, and that's only about 1 centimeter across on a Petri dish versus the wild type, that's 15 centimeters. And our whole genome sequence analysis using this in silico comparative approach allowed us to identify Compact here on linkage group, I don't remember. So we actually went after 18 different classical mutants, and these were typically in phenotypes that were considered to be valuable for either using the system as a model organism for biomass deconstruction or directly for industrial or biomass processing. So Compact, and this shows the overall morphology, and then this is the wild type under a microscope, and this is a mutant called Tangerine, which has a hyperbranching phenotype. So one of the things that made this more straightforward is that we had good genetic information. So in some cases, we were only looking in about 80 kilobases of DNA to find that particular mutation. So we didn't have to look in other chromosomes. We didn't even have to look beyond the flanking markers. And we used what we call in silico subtraction. So you compare all the genome sequences to the reference genome, and then you look and see what polymorphisms are shared. So there was various levels of polymorphisms, from a high of 140,000 to a low of about 20,000. And the thing that was really powerful was that most of the polymorphisms that we found were shared among multiple strains. And so that allowed us to ignore them in our analysis. So you can see we had a high of about 20% and a low of 2% unique SNPs. And this just shows how these polymorphisms were inherited. And what we could see was very much the fingerprint of the historical legacy of the Fungal Genetic Stock Center collection. Most of the polymorphisms were shared, and we could attribute those shared polymorphisms to different strain lineages, so the different origins. So 7035 was most highly related to the reference genome strain, and strain 1363 down here was very different from the reference genome strain, but it shared characteristics with other strains. And you can see the size of these haplotypes varies in regard to how closely related the strain is to the reference genome strain. So we were able to identify these mutants with a high degree of certainty, and what we often found was using the gene deletion mutants, we could recapitulate the classic mutant phenotype in the gene deletion mutant, although not always. And one of the things I just want to mention is that there's a lot of off-target information here. And this is an effort I've made in advocating that people not throw away or ignore their off-target information. So this is, molecular bycatch. And what I'm showing here in my Bart Simpson hair graph is the size of insertions and deletions in the whole genome sequence. And this is specifically the size of insertions and deletions in the coding sequence of genes. So not surprisingly, they're much more likely to be 3, 6, or 9 bases, and not likely to be 2 or 4 or 5 nucleotides in length. And that means that this is actually protein structure data, just waiting to be analyzed. And it's similar for single nucleotide polymorphisms. It's like a mutant scanning analysis, just waiting to be analyzed. Similarly, we looked at the mitochondrial genome in the DNA sequence that we had. Mitochondrial genomes have been characterized a little bit, but not quite as extensively. And the importance of this is that mitochondrial enzymes are often the target of agricultural fungicides. So the plasticity of mitochondrial genomes is important when you're considering the ability to use agricultural fungicides on a large scale. So the success of this has led us to use whole genome sequence analysis to characterize, and I love to say this, all of the remaining anonymous mutants in the Fungal Genetic Stock Center. So this is adding value in a tremendous way. There's about 400 anonymous mutant loci, metabolic mutants, sexual reproduction mutants, morphological mutants, temperature-sensitive lethals, regulatory mutants, mitochondrial mutants, and then we also invited the community to nominate strains. So several people had, for example, projects that they had abandoned in their, excuse me, as undergraduates or graduate students because they never got close enough to identifying the gene. Why is the DOE interested in this? Well, this picture shows Neurospora, it's the fuzzy orange stuff here, under the bark in a pine tree after a fire. And then just in 2014, it was demonstrated that Neurospora can be an endophyte and a plant pathogen. This is from Nature Scientific Reports. It shows Neurospora killing a pine seedling Positive control is Heterobasidium monosum killing a pine seedling, and a negative control is a healthy pine seedling. So with the DOE Joint Bioenergy Institute and the JGI, we've already sequenced over 550 genomes, and that's presently in analysis. So I think I've shown you that historical genetic collection has a new relevance in a modern era. Fungicide resistance, for example, biomass deconstruction, plant microbe interactions, and this is useful to the broader plant pathology and the phytobiome community. The collection includes plant-associated and plant pathogen strains. We've used various levels of DNA sequence analysis to characterize genetic and phenotypic variability. And the in silico comparison allows us to identify these anonymous classical mutants very rapidly. Lastly, the off-target sequence data has value for protein structural and biochemical characteristics. So I'm very fortunate to have found a home at Kansas State University, and my participation in this forum is supported by the NSF RCN grant for a community of ex situ microbial germplasm repositories. Thank you very much.
We'll have a panel following the speaker, so there'll be a chance for questions then. Our next, oh yeah, thank you. Our next speaker is Marcia Maues, and she's from Embrapa in Brazil.
Good morning. Thank you for the audience. Thank you for the invitation, David and everybody here. Well, I'm back. I'm going to talk again about pollinators. We had a wonderful talk yesterday, and I'm going to talk a little bit more. Some things will be maybe repeated. I'm sorry. But, well, pollinators and food security, risks and opportunities. I come from Embrapa. I showed you yesterday this picture. This is where I live, where I work, in Belém, Pará. It's in the north of the Amazon, of Brazil, in the mouth of the Amazonas River. My institution is the Center for Agroforestry Research of the Eastern Amazon, belonging to the Embrapa, which has the short name, Embrapa Eastern Amazon, and the acronym CPATU, because it reminds to another name we had, but we keep the acronym. It's located in Belém, Pará, in the north of Brazil, and it was created in 1939 as the Agronomic Institute of the North. And when Embrapa was created, it was incorporated to Embrapa. So we have a long history in the region. Well, I'm talking again about the world of pollinators. We've seen a lot about pollinators in this meeting, and animal pollination, it plays a vital role at regulating ecosystem system in service in nature. So pollinators are everywhere. Everything we eat, most of the things that we eat, at least 3/3, we have to thank to our pollinators because it depends totally or partially for animal pollination. We had a pollinators world agenda that started in the mid-90s when scientists and agriculturists around the world became concerned by the decline in pollinator diversity. In order to sustain pollinator services associated with agricultural systems, the international pollinators initiatives were created in Canada, North America, Europe, Asia, Africa, and Brazil, following a big meeting that was held in São Paulo in 1998 that was called the São Paulo Declaration on Pollinators. In this meeting, some issues were pointed out that should be addressed in order to understand the relation with the pollinators and food production. It was important to monitor pollinator decline, its causes and its impact on pollination services, to address the lack of taxonomic information on pollinators, to assess the economic value of pollination, the economic impact of the decline of pollination services, and to promote the conservation, restoration, and sustainable use of pollinator diversity in agriculture and related ecosystems. So it was the beginning of a big world campaign to understand and to study more the pollinators. One of the most active is the United States in the initiative that has been producing many beautiful information and very important publications. The pollinator-dependent crop, the crops which are pollinator-dependent on animal pollinations vary to many degrees. Some are totally dependent, some are partially dependent. But 5 to 8% of the current global crop production is directly attributed to animal pollinations. We have seen previously that there are 4 crops that are the basis of calories and human food that does not depend on pollinators. But our life would be very, very boring if we only had these foods. We need vitamins, we need other food, we need fruits, we need vegetables, and many of these are here because of the pollinators. Otherwise, we would have a very restricted diet, and it would imply in human health and other things. So, the conservation of pollinators in cultivated areas is essential to ensure a good harvest. And also, the same thing is valid for natural areas, because we also rely on many harvesting products from the forests everywhere. Land use changes, habitat loss, and landscape fragmentation is one of the major risks for pollination and pollinators. The removal of natural vegetation by deforestation, the conversion for agriculture, infrastructure, and new urban projects reduce the availability of food, nesting sites of bees, and may hinder their mobility by lack of connectivity. When we talk about pollinators, we are talking about a large variety of animals. But bees, as we always hear about bees, is because they are the major pollinators. They are the most, the majority of the pollinators are bee pollinators. So the pressure by opening new agricultural areas as compensation for the deficit of pollinators increase the habitat fragmentation, affecting negatively their communities. So when you have an area that is being cleared for agriculture, for other projects, we are removing the natural areas, and we are removing the nesting areas for pollinators, and the alternative resources they use for living. Another risk is the environmental pollution. The air pollution impedes bees' ability to find flowers. There is a study that was made that shows that the pollution interferes with the ability of bees and other insects to follow the scent of flowers to their sources, undermining the essential process of pollination. So air pollution, environmental pollution is another major risk for pollinators. Intensive agriculture management, monocultures, is also a major risk because it also removes the natural areas and make it difficult to the pollinators to survive. The intensive cultivation of the soil, the use of agricultural machinery which revolves the soil and kills many nest-grounding bees, the herbaceous, eliminate the herbaceous vegetation, and the introduction of genetically improved plant varieties reduce the amount and varieties of alternative food and nesting resources for bees. A few species are able to recover after disturbance, like said in this paper that was published in 2005. Another serious risk is the indiscriminate use of pesticides. The applications of pesticides to control pests and pathogens with high toxicity to pollinators and without observing their patterns of visit can lead to death, act as a repellent, and also cause sublethal toxic effect, flight disorientation, reduction in the production of offspring, and other factors. It has been discussed a lot, I will talk a little bit later, about the colony collapse disorder, one of the major drivers of colony collapse disorder might be the use of some kind of insecticides, pesticides that are causing bee disorientation. So the use of pesticides can suppress or reduce the production of nectar and pollen in some plants, reducing the supply of food for pollinators. Another point that is very risky for pollinators is the global climate change. Sudden changes in meteorological phenomena may change the geographic distribution of species of bees and plants. As Professor Inoue has pointed out in his studies, the mismatch of flowering plants and the pollinators that are hatching in the area, they may occur in different periods, as the climate change can change the period of flowering of some species. So this will be also a conflict for plant-pollinator interactions. And also the timing of environmental events attributed to climate change shows that it affects pollinators, herbivores, and seed predators as well. This mismatch and activity decreases the diet breadth of pollinators. Another problem is the introduction of exotic species or invasions. The exotic pollinators that were introduced in some countries to pollinate tomatoes in protected cultivation, like the bumblebees, they became a problem when they escaped from these protected areas and spread to the nature. They were competing with native species, they were mating with these native species, and they're producing hybrids that were not able to survive, and they were not good pollinators for native crops. They were good pollinators for protected cultivated areas, like tomatoes, but they were not good when you introduce, and these bees are able to fly away and start new colonies. We have other habitat alteration and invasions affected by pollinators to the same magnitude by decreasing visitation rates. The vertebrates that altered landscapes and insects, excluding bees, in invaded areas were the most affected pollinators taxa. The study made by Monteiro and Castanho on the impact of landscape alteration and invasions to pollinators shows this relation, that the problem of invasors and the pollinator in the areas. Another problem with the pathogens and parasites in bees mainly, we have the varroa mites, the nosemoses, the American fullbrood, and the chalkbrood, and others. And they all are problems that affect the pollinator's health, especially on bees. So all these problems that I'm showing you came to another point, that is the colony collapse disorder, that started with some reports from apiculturists in 2006. They said that the bees were vanishing from the colonies. They were not coming back. The bees that went to harvest, they didn't come back to the colony, and the colony was left alone with the queen, with brood, with honey, and nobody understand why, what was happening. So the colony collapse disorder is a phenomenon that occurs when the majority of the workers of a bee in a colony disappears and leave behind the queen, the plenty of food, a few nurse bees to care for the remaining immature bees, and the queen. Once thought to pose a major long-term threat to bees, reported cases of CCD have declined substantially over the last years. During this time, from 2006 to now, many factors were pointed out as causes of the CCD. And it's believed now to be a complex and a result of multiple factors. A study that evaluated 61 factors found that no single stressor stood out as the main cause of CCD. This study that was published by Dennis van Engelsdorf and collaborators, they show that there is a range of factors that is affecting the colony collapse disorder. So one of them might be, okay, 3 minutes, might be the use of pesticides, the indiscriminate migratory apiculture, and the problems in bee health and other problems. But we have also other factors that are good for pollinators. The indigenous and local knowledge about pollinators, they are being, how can I say, not taken into consideration. The practice that included traditional farming systems, maintenance of diverse landscapes and gardens, kinship relationships, and protect specific pollinators and cultures and languages that are connected to pollinators should be addressed and should be taken into considerations. The pollinators, especially bees, were very important in ancient cultures, and they are a symbol in many civilizations. And they are globally significant heritage sites and practices, the symbols to identify aesthetically significant landscapes and sacred places about peace occurs in all major world religions. To mitigate these impacts, the adoption and measures to promote the attractions and the conservation of pollinators in cultivated areas, there is a group of factors that is called the pollinator-friendly practices. The pollinator-friendly practices are measures that you can take in order to keep the pollinators in the gardens, in the crops, and to avoiding removal of the soil, removal of the soil, tillage, keeping areas and forests and secondary vegetation, offering places to nest for bees, encourage the adoption of agroforestry systems that utilize not monoculture, but when it's possible to use mixed cropping, to maintain the connectivity of reminiscent of forests and natural vegetation, making ecological corridors, and to promote the importance of friendly practices among agricultures and the general public. The corridors to help to maintain and recover the degree of cohesion of otherwise fragmented ecosystems, and these corridors would enlarge the habitats and help the dispersion of young animals and reuse of empty habitats. The ecological intensification and farming would mutually beneficial pollinator, diversity, and crop yield outcomes in small and large farmers. Ecological intensification or the improvement of crop yield through the enhancement of biodiversity may be a sustainable pathway toward greater food supplies. In the last 10 years, many books were published, and this is good for the public awareness about pollinators for different publics, for students, for professionals, and for the general public also. And in Brazil, we also had a lot of new books published about pollination and pollinators. The FAO's Global Action on Pollinator Services improved the participations in events at national and international levels, the provision of support and liaison with regional initiatives, the African Pollination Initiative, for example, addressing climate change and pollinator services, awareness, and developing and providing tools and guidance for documents. There is a big project that's finished last year, the Global Pollination Project, that was in between 6 countries, Brazil, Ghana, India, Kenya, Pakistan, Nepal, and South Africa. And all this project gave many information about pollinators and pollination. In Brazil, we had 7 projects on important crops for Brazil, growing from the south of Brazil to the north of Brazil. And IPBES, that Dr. David Inouye spoke yesterday, was the assessment on pollinators and pollination and food production. It has a lot of recommendations that is being published now. We had many initiatives in many countries also. There is a campaign in Brazil that's called the To Be or Not to Be, that it stresses and shows the importance of pollinators to the fruit production, and mainly the fruit and crops. And now here in United States, the President Memorandum signing for pollinators, it was signed in June last year, the strategy to promote the health of honeybees and other pollinators in order to reduce honeybee colony losses to economically sustainable levels, to increase monarch butterfly number to protect the annual migration, to restore and enhance millions of acres of lands for pollination through combined public and private action. And initiatives to make gardens for pollinators also, and to create pollinators and native plant gardens to improve the pollinations in areas close to cultivated areas. The pollination also benefits society by increasing food security and improving livelihoods. The conservation and sustainable use of pollinators intersect on a number of key issues. Pollinators contribute to biodiversity and life on Earth, food security, and to the global economy. Thank you very much.
All right, so I meant to remind the panelists when I came up here before as to who you were, so you'd be paying attention, but hopefully everyone was. So if Lisa and Steve could join us for the panel discussion, in addition to Marcia and Kevin. Yeah, I knew. All right. Yeah.
Shall we first do a quick introduction? The two speakers maybe don't need to introduce yourselves again.
Hi, I'm Lisa Castleberry. I'm a research mycologist with the USDA Agricultural Research Service, just located across the street here in Beltsville, also director of the U.S. National Fungus Collections.
Does this work? So my name's Steve Young, and I have two roles. First, I'm a weed ecologist at Cornell University, and I also direct the Northeastern IPM Center. And so I know there was a lot of discussion, or there's been some discussion about that, so I'm particularly interested in that, but I guess I'm the only one that has to introduce myself, so. Weed ecologist, WEED, yes. Sorry, integrated pest management, yes, I assume, but yes, good. My name is Steve Young, yes.
So having been in the audience for the last panel, I would just like to say something to the panelists. When you speak to the back of the room rather than to each other, because often the panelists, it's very hard to hear you because you tend to. So you have to pretend like you're talking to each other, but talk loud enough that you can reach the back of the room. Are there any comments that you would like to start with before we open up to questions?
So I had a question for Marcel. So see the colony collapse disorder, I saw one of your slides said it doesn't occur in Brazil. So can you expand on that?
What we know about colony collapse disorder in Brazil is that there are some reports about bee mortality, but very localized, very specific places when you have an incorrect use of pesticides. But what people say is that the Brazilian Africanized honeybees, they are not so. I'm sorry. Yeah, we don't have the problem as migratory apiculture. We don't have our crops are not so. The pesticides are not used in so extensive areas, and there will not have any how can I say, the problems that occurs here that were reported in the United States and Europe, we don't have in Brazil yet. So there is a recent, was published this year, the study that says that the problem in colony collapse disorder was not reported in Brazil yet.
I'd like to add that in that picture I showed you of Neurospora sporulating on a pile of sugarcane bagasse, when Neurospora is, or when sugarcane tailings are used as fertilizer, they spray them on fields, and then the Neurospora will sporulate across the field. bees will collect Neurospora canidia rather than pollen, just because of its tremendous prevalence in the environment. I asked David yesterday, and he said they're probably just feeding it to their young. There's no report of Neurospora being a problem in apiculture.
Now I had a question for Kevin about the phenotype data that's being collected. How are those data just appearing in journal articles, or are they being databased along with the collection information so that it's also searchable?
So there's two levels of information on this. The classical genetics data from the Fungal Genetics Stock Center and from the Neurospora research community was assembled into compendia beginning in 1986, recapitulated in 2001, and hosted online in the Fungal Genetics Stock Center database thereafter. And now it's being managed by a retired Neurospora geneticist at Leeds University in the UK. So that's the classical phenotype data. So things like that morphological mutant. A lot of the biochemical mutant phenotype data isn't really interesting. It either grows or it doesn't grow. And, some of those phenotypes are kind of relegated to the classical genetics era. The morphological things are more interesting. I should add that the demonstration that Neurospora can kill pine seedlings was independently recapitulated, and it was shown that a single gene mutation was enough to switch Neurospora from its endophytic to its pathogenic phenotype, kind of reestablishing the value of these classical genetic traits. The Neurospora functional genomics program, through which we generated, or the mutants were generated at Dartmouth and at UC Riverside, and they were sent to the Fungal Genetics Stock Center, where we preserved them and arrayed them in 96 well formats for distribution, either as individual mutants and as an arrayed set. That included an extensive phenotyping program where single gene deletion mutants were phenotyped by undergraduate students at UC Riverside, at Texas A&M University, and at a number of other institutions that had small contributions. Those phenotype data were uploaded to the Neurospora genome server, then maintained by the Broad Institute at MIT as part of the functional genomics program. We advocated that those data would be published in the boutique journal that the FGSC had published since 1960, now called Fungal Genetics Reports. But those data were never published, and when the functional genomics program grant from the NIH ended, the Broad Institute turned off the server, and those data are no longer available, and which is, I think, a tremendous tragedy. We have the mutants, and the phenotype data was generated under NIH grants, but the data are no longer publicly available.
I was just going to ask you a question, Steve. So if you were going to follow up on Kevin, you should.
So this wasn't really a follow-up per se, but Kevin, from what I gather, it sounds like your organization or group or center or whatever you're actively doing things with your collections. You're actively looking at things. So our center, we have a collection, if you will, and we do the same thing because we fund research, we fund grants. But I'm just curious to know, are there others external to your group that are doing the same things you are, or are you kind of leading in what you're doing with your collections?
Thank you for that. I want to correct a misapprehension, a misconception. The Fungal Genetics Stock Center consists of myself and one research assistant in shared laboratory space. What we do on a shoestring. And when we had NSF funding, we had an outreach mandate, and that included bringing students into the lab. We no longer have that mandate, and everything we can do just to stay afloat is supported by Kansas State Department of Plant Path. We support a diverse an extensive research community. So Neurospora is used by several hundred laboratories around the world, and we provide materials to those researchers at a small cost. When we moved, we were obliged to raise our fees. The NSF-supported collections have always been encouraged, required, obliged to charge small fees. We raised our fee from $20 to $50 and saw a 50% decrease in orders from our collection. And I view that negatively both from the collection perspective. It limits our ability to be self-sustaining, but also in that it decreases the ability of the community to use validated materials. It encourages peer-to-peer ad hoc exchanges where, you know, the newest grad student in the lab is sent to the back of the freezer to find the strain that was published 10 years ago. And there's no validation, there's no, you know, quality control on that. They're often distributed without regard to shipping regulations. You know, Neurospora is still considered safe to ship even for the GMO, but for plant pathogens, you know, is there any APHIS people here? You know, if someone carries out research with a GMO plant pathogen, which they obtained from a colleague without regard to permitting, and then they publish on it, either they're, you know, fraudulently committing research or they're violating permit requirements. So it's kind of a scary circumstance for me. The short answer to your question is that we do what we can with a very small number of people, and we support a diverse research community, not just Neurospora, but plant pathogens, Aspergillus, Fusarium, a wide number and a growing number of organisms.
Okay, that begs the question. You aren't required to make sure people have permits before you send them things?
We absolutely do. And this has been, I think, you know, the fact that we require permits and peer-to-peer exchanges can often be conducted without those requirements puts individual researchers at risk. So we have 49,000 tagged integrant mutants of Magnaportha, which we've never been able to distribute because no one's ever gotten a permit until this one researcher got a permit to receive the entire collection from us. Permitting requirements, you know, I advocate that culture collections be associated with APHIS, be integrated with the permitting system so that we can provide expedited permitting for people who want to receive materials for which permits are required. That would facilitate the process of obtaining materials with the appropriate permitting requirements fulfilled. As it is, it's easier to get things on a peer-to-peer basis, or quite frankly, to go out and re-isolate the organism and key it out yourself, which then, you know, you're not doing a direct comparison. You're, you know, I like to use the example of apples to apples comparisons. And we have all these beautiful graphical images on the walls here. You know, when you go out and isolate your own version of the same species, you may be doing a Pippin to Golden Delicious, or you know, a Brayburn to Red Delicious comparison. It may be apples to apples, but you know, it's not the same. These are cultivars, but it's not the same variety. So I really advocate that permitting be integrated into collections so that people have a good way to get things under appropriate conditions.
I just have one sort of follow-up on that in terms of, so you're operating on a shoestring and you have all of these cultures. Are you the only authoritative source for all of these cultures, or do you tissue bank? I mean, what mechanism is there in place to ensure that this resource is still available?
That's a really valuable and important question. We are the sole source, the sole authoritative source for most of these materials. Many of the, for example, the reference genome strains are available from a number of different collections. I mentioned yesterday that I have a twin brother. So, you know, there's this bizarre presumption of equivalence about strains that are banked in different collections. The straininfo.net resource will show you the history of strains. Also, the American Type Culture Collection will show a brief history, an abridged history for materials in their collection. It indicates who the depositor was and sometimes where they got it. That being said, one of the efforts of the U.S. Culture Collection Network has been to liaise with the U.S. Department of Agriculture National Laboratory for Genetic Resource Preservation, and through these efforts now, 8 major collections, including most of the strains in the Fungal Genetic Stock Center, are backed up offsite at the Fort Collins facility. We had originally sent the gene deletion mutants as an arrayed set in 96 well plates, and just this calendar year, we backed up our historical classic mutant collection at the USDA facility. So even if the FGSC suffers a catastrophic loss, our building doesn't have backup power, the materials will still be available.
So, because this session is stressors and biological systems, I guess I had a maybe question for both of you, and maybe this is larger perspective of, you know, what are the, obviously there's climate change. It's a stressor that's, you know, becoming more of a factor on pollinators or you know, Kevin, or what you're doing is that in general what you're seeing, or are there other stressors that have always been there that are equally, you know, as important? And I'm coming at this from my background is weeds, so I'm looking at what are the stressors on plants, but I would be interested to hear, you know, and some of the stressors that you're seeing, or that, you know, is becoming more important as we move forward here.
One of the areas of growth in our collection is endophytic fungi that establish mutualistic or beneficial relationships with plants. Other collections deal with plant-associated bacteria, especially the USDA NRL collection. There's been a tremendous growth recently in the demonstration that microorganisms can have a beneficial impact on plant growth. So from the tripartite system described by Rusty Rodriguez and his colleagues in Seattle, where a plant that has an endophyte, a fungal endophyte, can grow at 65 C, while neither the plant nor the fungus can grow at that high temperature. And it's tripartite because the fungus actually has a viral infection. So you have to have all three. And when you have all three, this plant can grow at 65 C. So that's global climate change right there. Other fungi have the ability to influence plant growth, where the Noble Foundation is working on subaceanoid fungi that allow perennial grasses to grow more robustly. So these growth-promoting organisms are an important area of growth for our collection. And I guess it was Dr. Wotecki yesterday mentioned the AfloSafe system. AfloSafe is a non-toxin producing Aspergillus fungus that you can apply as a granular treatment to your field, and it prohibits or precludes subsequent colonization of that plant by toxin-forming fungi. AfloSafe is available as a commercial product, and our collection has received a number of strains that are useful for that application, and I think it's important that those be available from public collections so that they are not all behind a paywall. Especially in regard to the Nagoya Protocol, different strains may be necessary in different countries and different geographic regions, and the availability of those strains through public culture collections ensures that those organisms are available to farmers at every level and not just on a for-profit basis.
Well, concerning the pollinators, one of the major threats, as I showed you, is fragmentation, habitat fragmentation. When you have the habitat fragmentation, you lose areas, you lose natural, the species that pollinators depends upon, and you isolate areas too. So it's very important to have, to restore these areas, to have ecological corridors to link again the fragmented areas, and to there are efforts that are planting plants to attract the pollinators, to keep the pollinators in cultivated areas, close to cultivated areas, because these plants are dependent on the pollinators, and they don't, they cannot live in the area when there is no flowers, there is no food resources for them, there's no places to nest and everything. And the climate change is also very important because, as I told you, it might lead to a mismatch between the flowering season and the occurrence of the pollinators in the area. So when this happens, you will have there's a combination that will not work. So climate change is being studied for long-term studies that showing that in phenology there has been a difference. Some plants are flowering with a delay or earlier, So it's not combining with the pollinator activities in the area. There are many other factors, and the indiscriminate use of agrochemicals when you don't have the proper care to use the agrochemicals in the period that the plants are not flowering, or when you use in agrochemicals that are not compatible with the pollinators, they kill everything. And now we had a problem with this Zika virus, the mosquito that is being spreading, that's coming from Brazil. And is going in areas that it was not occurring, and some people had the brilliant idea to spray insecticides to kill the insects, and it was not insecticide that was killing only the mosquito; it killed everything. So there was a big mortality of bees in some areas, and there are things like this that is happening that we have to take care and protect the pollinators, but have to think about in a very holistic idea on how we can protect the plants, the crops, and the pollinators that depends on this, the crops that depends on these pollinators. It's a very delicate balance. And for vertebrate pollinators, for other pollinators that are not insects, it's more difficult. It's more complicated and delicate because they depend on natural areas. They depend on natural vegetation. And when you talk about bees, you have the apiculture, you can raise bees, you have the stingless bees that you also can raise bees, you can manage solitary bees, but for other kind of pollinators, for example, weevils, we have the pollination of oil palm that is completely dependent on weevil pollination. In oil palm in Brazil, in Africa, and in Asia is an important crop. But in Brazil, it has a problem with a syndrome called the yellowing that kills the plants. So they developed a hybrid that is tolerant to this yellowing, to this problem, but it's totally dependent on assisted pollination. So people have to hand pollinate all the plantations, and the cost is about 1/3 of total production. So in Brazil, the oil palm is an important crop, but it's under threat because it has this syndrome that is killing the Ileis guineensis, which is the African oil palm. We have a Brazilian oil palm. It's not very productive, so they developed this hybrid that is tolerant to the yellowing, but is totally dependent on pollination. So they have to come back, the geneticists have to come back to the lab and try to find a way to keep the ability of the hybrid to attract pollinators, because it doesn't attract pollinators. It has, it's a very, very interesting thing, because in oil palm, there is a blend of scents that attracts the pollinators, which are very small weevils. And you see in the inflorescence of oil palm, it's a monoecious plant, the female's inflorescence and the male's inflorescence. The male inflorescence attracts over 20,000 insects of these species, the species that pollinate, which are basically 3 or 4 species of a weevil. And they go in the female inflorescence by mistake because it has the scent, but it doesn't have any resource to offer to the pollinators. And by mistake, they pollinate, but the hybrid didn't keep this characteristic. So there is no way that they can attract the pollinators, even if they are cultivated side by side. And so what the companies have to do, they have to cultivate the African oil palm, they have to take off the pollen grains, prepare the pollen grains, and they have human pollinators to make the pollination process. So this is still worth, because it's very valuable crop, but it's a big problem.
What has been the impact of the Africanized bees in Brazil on pollination?
Well, Africanized bees we didn't have the honeybee in Brazil, so they pollinate also other plants that are not the exotic plants like apple. They're being used for apple pollination, and the impact in the native plants. Some studies that were made that show that there's no huge impact. All the plants that I've been working with, that I have worked with in the Amazon region, they were not pollinated by honeybees. Honeybees were not a problem nor a benefit to the plants. For example, the one I showed yesterday, the Brazil nut flowers. The Brazil nut is totally dependent on pollinators, but the flowers has a very peculiar structure that honeybee cannot open the flower to access the resources. The Brazil nut flowers, they need strong bees, like the bumblebees, carpenter bees, that are able to force the entrance and go into the flower, inside the flower. The honeybee doesn't make anything. So in general, it pollinates plants that are exotic, that were cultivated in Brazil, but they're not native. And they are not a big problem. They are very good for honey. They are good honey producers, but not a problem for pollination.
What is the impact of invasive species on the pollinators?
Some invasive species, for example, we have the bumblebees, which in Brazil is not a big problem because we don't have, we didn't imported the honey, the bumblebees to pollinate. Like Chile had the problem, Argentina had the problem, but in Brazil we didn't. So invasive plants sometimes can be with poisonous nectar or things like this, but it's really not a big issue in Brazil, the invasive.
I'm going to answer that one a little bit, not so much for Brazil, but at least in North America, often invasive plants are beneficial to honeybees because they're fed on by honeybees. So the honeybee is not native to any of the Americas. So right, they're a problem to the native plants, but they're not a problem to the bees. In fact, beekeepers sometimes try to get certain plants like yellow star thistle, and another one is yellow clover, off of the noxious weed list because they want that plant to be there. Yes, yes. There was some initial theory that what made a plant invasive was it didn't need a pollinator, but that wasn't found to be true.
This goes to Marcia. Are there any current ongoing plans to exploit the few resilient bee species?
I'm sorry, I didn't get the question.
Are there any ongoing plans, any actions to exploit or harness the resilient bee species? You did mention in your presentation that there are some bee species that are resilient to stresses.
Resilience to stressors. I would say that some native bees, they're very resilient, very they can adapt. As I said, that when you're talking about agriculture and you have an area that is being you applied tillage, when you're revolving the soil, removing the soil, and you're distracting the nest for ground nesting bees, they are very, very sensitive to it. But other species that nests in trees or make other areas like decaying wood, they are more resilient to the agriculture. For when you talk about soil in the agriculture, when you are talking about removing the soil and revolving the soil before planting. I'm sorry?
So you say some of them do live in certain decaying trees, right?
Mm-hmm.
How are you using that knowledge to help agriculture in Brazil?
We are recommending for example, when you have, talking about passion fruit is pollinated by carpenter bee, and carpenter bee make nests in decaying wood. In areas, when you see in the north of Brazil, we still have lots of natural vegetation. It's not a problem for passion fruit. But in the southeast of Brazil, where passion fruit is an important crop and there is no natural vegetation around, passion fruit pollinators is missing. They have to hand pollinate, or they have to create how can I say, to give opportunities to the bee to colonize again, offering decaying woods, offering places for nesting, offering sometimes even you put some trap nests, some bamboo nests or wood blocks in order to help these bees to make nests in these pieces of wood, because they are not available anymore in the nature. I don't know if I've answered your question.
So I've always been interested in the honeybee issue, having come originally from California and going home all the time. And it was a curious thing just thinking about when the bee collapse started to happen in the U.S. Was at a time of peak activity of bringing bees from bee colonies from all over the U.S. through the Central Valley of California. And at the same time, there was a peak in putting in new areas of breaking new ground in California, and in an area where we saw a lot of valley fever, which is a fungi. And I made the connection, and I may be, faulty because of just minimal information, but there was a possibility that the combination of that fungi and bringing the bees from all over the country through California and then back out again could be spreading at least a related fungi. We did in the soil survey, we did an analysis of habitat for the valley fever fungi and hit it right on as to the areas where we would expect to see the fungi and where it was actually being plowed up. We also had a huge increase in valley fever in the valley, partially because they were opening up these areas to agriculture that hadn't been before, right up into the foothills. So it's all a combination of different pieces of information, place-based. Obviously, it's very simplistic what I observed, and it was never substantiated by real bee people. But I think that's always the interesting thing about combining different pieces of data, place-based, and outsiders taking a look at things. It's more or less a comment, but I'm just curious whether there was any collaboration between the fungi-based folks and the bee folks, the pollinator folks, as to whether there was a connection for the collapse.
Since you asked specifically about California and North America, I'm going to let Kevin Hackett take first crack at answering your question. Is this on?
Hi. So a combination of things. One thing is that colony collapse disorder, when it really hit in full force here, but it's disappeared. It itself has disappeared. We don't really have colony collapse disorder in the country anymore. The problem is, however, that we're still losing about the same number of beehives. So colony collapse disorder itself in this country, one of the theories is that it was a new virus that passed through, and that the virus was a very virulent form of the foreign wing virus, which was in varroa mite. And one of the reasons that Brazil might not have been hit so bad is because the Africanized honeybee is more resistant to varroa mite. So therefore, it might not have spread the deformed wing virus, which again is a more virulent form of that particular virus. Well, what happens when a virus gets into a bee population is that it's going to be self-selection. So the bees that survive are the ones that are bred. So it could be that we bred our way away from colony collapse disorder. But the problem is we still have the loss of bees because we're still getting a total bee loss of about 40% a year. So there's a lot of factors that are killing bees. As far as a fungus, I don't know. It could be one of the many factors. We know that fungicides potentiate insecticides or miticides. Miticides, by the way, you're using in beehive to protect the bees from varroa mite, but fungicides can potentiate miticides and insecticides by 2,000-fold. So where there's some fungal problems, there might be more fungicide use and therefore more synergistic kill of honeybees. But I have a question too about about Brazil, and that is whether you have any germplasm efforts to breed bees, gene banks or whatever in Brazil, and if so, what kind of records might be available?
Well, I couldn't say for sure, but the gene banks in Brazil for bees, at least the one that I'm working with, are the collections in the museums. Bees are deposited in museums. Probably people from Ribeirão Preto have some information on breeding. The people from Spencer, you know Spencer from Ribeirão, Leonel, Gonçalves. So I'm not sure, but probably they have. I'm sorry if I can reply completely.
This session is about biological stressors, which is a very, very broad topic. Stressors and AIDS. So we've heard about pollinators as AIDS. We're thinking about fungal genetics as sometimes it's AIDS, but it is a control agent as well as being tested. I want to ask a very general question. Many people in the group may not be aware of the International Treaty on Plant Genetic Resources for Agriculture, and it's this amazing, unique agreement among CBD countries, not all of them, but most of them, for the open exchange of plant material in about 60 species that are crop related for research and plant breeding. So there's this enormous number of samples being moved around the country. This highway is seamed and cuttings without restrictions on permits, except for phytosanitary. But it's like a super highway of material. The information is not moving as much as they're not digitizing the accessions completely, so the information is not moving, but the material is. Ironically, all the material related to security on the stressor and aid side, insects, pathogens, parasites, fungi, all that stuff is not covered by the plant treaty. All of the restrictions of the Nagoya Protocol and national permitting apply, so there's no superhighway for that material. I find it ironic because yes, there is money to be made in providing control agents or replacement stocks of bees sharing. A lot like the sharing that Chris raised. When your crop fails, we help you with something that will work. Same thing for controlling pests or pollinators. What's your experience been about this? This is an issue that my organization has really pushed on in Nagoya. There should be, it help get language into the protocol for simplified activities. When is the non-commercial research that serves CBD's goal of sustainable biodiversity? Correct? What's your experience been
as we come across the breakthrough moment, relationship with agreement to solve this problem?
I would be very happy to know which are the 60 species that are, because in Brazil we have a very restrict legislation for biological. Yeah, because in Brazil we now have a new legislation, but we still are not allowed to use it because they are still making some files. So we have to register our research, what we're doing. So every native species that we work, we need a permission. And this is for all the areas from medicine to biology, everything. So there is I remember now with the Zika virus outbreak, Brazil had a very difficult, how can I say, it was very difficult to exchange material because the scientists were not allowed to send materials abroad. So they made a special legislation for this problem for Zika virus because it was a global threat. But for native species, yes.
Article that I spoke of with the simplified access in the Nagoya Protocol, the two examples were medical epidemic.
Yes.
And
Yes. It worked, but it was not so fast as people wanted. Maybe it took some months. Okay.
So the plant treaty has been hailed as a success, and it's described as being multilateral in that everybody agreed to the same terms. They set up a system for benefit sharing, and so if there's any commercialization, everybody has agreed to the same ground rules. By way of contrast, the Nagoya Protocol is described as bilateral. So every exchange is between the two country parties. So for example, many of us, when we were grad students, we did zoo blots, where you put 20 different taxa on a gel and you looked at the presence or absence of a particular characteristic. If those isolates came from different countries, you would have to get prior informed consent on mutually agreeable terms from every country. The Nagoya Protocol has led to significant uncertainty because of this bilateral nature. For microbes, the World Data Center for Microorganisms is establishing a framework that will serve as the access and benefit sharing clearinghouse for the Nagoya Protocol. And Nagoya is supposed to work through this access and benefit sharing clearinghouse. Nagoya was ratified in fall of 2014 and went into effect shortly thereafter. To date, only a few countries have established access and benefit sharing clearinghouses. Brazil is a tremendous example of a country that has been proactive in this regard. The EU has established a framework that most of the member countries have agreed to. One of the very frightening things about Nagoya is that a country can establish ex facto regulations, so they can say everything that was ever isolated from our country is subject to access and benefit sharing, while formally the CBD went into effect in '93 and Nagoya in 2014. The EU legislation, for example, says no EU country should establish ex facto legislation, so that everything that was accessioned into public culture collections or other kinds of biological repositories before 1993 is still considered to be the shared heritage of humanity. And things after 93 are the property of the country of origin or the party of origin. Brazil, most recently, in their legislation that was just approved, says information in genome sequences in public databases is subject to access and benefit sharing regulations. So I would say you can't put the genie back in the bottle, but Brazil's trying. And I've just returned from a workshop of the World Data Center for Microorganisms, where we had significant discussions about the impact of the Nagoya Protocol on the exchange of genetic resources. And the point was made that if you have a cosmetic or a medicine or a food product processed food that contains resources from multiple parties. Under Nagoya, each party can say we want 1%, and so pretty soon you have you've consumed any possible profit. So the companies are very frightened about this. On the positive side, I would say that if I mentioned these AFLA-safe products, the fungi that you can apply to a crop to prevent the development of toxins subsequently, well, the best fungi are probably indigenous fungi. So that would encourage the researchers to go into the country where they're going to use that resource, where they're going to use that product, and identify native strains that are non-toxin forming and make them available in that country. And that's exactly what Nagoya wants to do. They want to encourage benefit. The benefits can be non-monetary, so they can be going into a country and establishing training, establishing a research facility, and that would satisfy the requirements in Nagoya. The other thing I'm going to say is that Nagoya, there's some really bizarre things. So you mentioned Zika virus. There's also an exemption for the pandemic flu preparedness project, so you can exchange resources for the flu vaccine development. It's specific to research and development, and so diagnosis isn't considered to be covered. So if you get, for example, if APHIS, the USDA Animal Plant Health Inspection Service, identifies pathogens on commodities that were intercepted at the border, they can do diagnosis on those, but they can't bank them and they can't do research and development on them. The weird thing is that taxonomy is not considered research and development, it's considered diagnosis. So you can do taxonomic characterization of materials that you intercede at the border, but you can't then subsequently develop them for a product. That's a great resource for identification.
More question going back to the basics, because I mean, when you want to act on biological stressors, public awareness is really important, and also decision maker. So I don't know if using the term biological stressors is more a US thing, or because when I discussed a bit with colleagues back home about that, they immediately understood more the medical term about stress, releasing adrenaline or health-related things, endocrinology, and actually when you talk to them about invasive species, pollinator problems with the bees, immediately it speaks to them. But if you use biological stressor, they have no clue what you're talking about. So I think it's important to define that for some of your community you want to outreach, because they directly link it to something medical, or maybe, I don't know, cattle that is stressed when it gets slaughtered or things like that, but not, they don't link it directly with the environment. So
Stressors
it's more
is
a term used amongst us, experts in it, but if you go outside, they understand something differently.
That's not a term that's really widely used in the plant disease community either. I think it's intended to include the fact that other things can impact plant productivity, such as drought, salinity, and other stressors such as that, abiotic stresses. I just want to get a plug in for the phytobiome initiative, which is being launched by the U.S., the American Phytopathological Society, and it's an effort to address all of the factors that influence plant productivity. So it includes abiotic stressors, it includes the plant microbiome, it includes things like arthropods, nematodes, even parasitic plants. So the Phytobiome Initiative is seeking to use a variety of approaches to double the plant productivity by 2050 to feed the future, and it's emphasizing things like non-chemical approaches, beneficial microbes, beneficial symbionts, and I encourage everyone to look at phytobiomes.org.
Can we give our maybe a hand? Great, thanks everybody.
So just before we break for lunch, I wanted to do another tally of folks who are interested in doing the tours tomorrow afternoon. So I know we've had some travel changes and that sort of thing. So can I a show of hands of who would be interested? So this will be around lunchtime, so around 1 tomorrow. Who would be interested in a tour here at NAL? Some hands. 3, 4. Okay. So the NAL tour is this building. It's yeah. So you'll have to go upstairs. Yeah. Pardon? Okay. All right. So okay, that's enough of a estimate. Okay, and then we also have over at the USDA facility across the street, which would be a 10-minute walk or so. So that's the aphid collection and the mites. Yeah. Tiny insects. So that's across the street. So who would be interested in that one? And ideally we'll have it so you can do both. We'll just put it that way. Okay. All right. Okay. Great. That's helpful. Okay. All right. So now is time for lunch, and we will get started again around 1:30. So thanks, everybody. Oh, sorry. I did want to say the wraps that are in the back are vegetarian. The other sandwiches are omnivorous. Okay. Very good. Thank you.